Somos una universidad católica regional. Más del 80% de nuestros estudiantes provienen del Maule y más del 80% de nuestros egresados se quedan en la región. Somos parte del desarrollo del capital humano avanzado. Fomentamos una universidad católica regional y generamos proyectos que van en directo beneficio de la sociedad. Provienen del Maule y más del 80% de nuestros egresados y el pensamiento crítico a través de nuestros planes formativos y sello valórico en pregrado, posgrado y educación continua. Somos parte de la historia de 15.000 personas que han pasado por nuestras aulas y hace más de 50 años que contribuimos con el desarrollo de la región, siempre responsable y comprometida con nuestro entorno. Contamos con programas de acceso inclusivo, porque creemos que no hay impedimentos ni sociales, económicos o físicos para concretar los sueños. La investigación que se genera dentro de la UCM nace desde las necesidades locales. La innovación busca cambios positivos en la sociedad, dando respuesta a los requerimientos de nuestra zona. La UCM es fiel a sus orígenes, pero mantiene su vista en el futuro. Somos tu universidad.
Good morning, good afternoon, everyone. Welcome to the workshop entitled Sampling Techniques in Extreme Environments, Sample Processing and Interpretation of Biochemical and Molecular Data. This activity is part of the International Biochemical Network for Understanding the Impacts of Climate Change on Coastal Whaling Ecosystem in BioNet. This project is funded by the Chilean Research and Development Agency, and the sponsor of this network is sponsored by the Center for Research and Advanced Studies, CIAM, from the Catholic University of Maule, which participate as the responsible institution, by the Center for Advanced Studies in Arid Zones, CEASA, as associate national institution, and by the University of Southern Denmark, who is the foreign partner institution. This network is confirmed by Dr. Sara Cuadros from the Catholic University of Maule, who is the national co-investigator, Dr. Aparna Banerjee, colleague at the CIAM and national researcher, Dr. Carlos Enriquez, the associate investigator, Dr. Botandro, who is the foreign investigator, and for myself, the principal investigator. The main goal of this network is generate and strengthen the scientific technological network between regional and institutions in Chile and Denmark, to understand the impacts of climate change on biochemistry of the coastal whaling ecosystem of the Eastern South Pacific, and considering the profiles of the investigators that confirm the network, this approximation will be done from an interdisciplinary perspective. So another important thing is that this project is anchored to the Climate Action Planning Program, CLAP. CLAP is a CEASA project that's trying to improve predictions of the biosphere component's response to climate change and its variability at multiple scales to support effective climate action plans. So in this context, the confirmation of this network will feed a regional ocean-atmosphere coupled model to understand the biochemical and molecular response of the marine environment to the projection of the climate change stressors. So now I will give the space for a brief welcome words of our Vicerrectora of Investigation and Postgraduate, Doctora María Teresa Muñoz.
Bueno, quiero darles la más cordial bienvenida a todos y todas a este workshop que está organizando el Centro de Investigación de Estudios Avanzados del Maule en donde nuestro investigador principal y que está a cargo de todo esto es el doctor Alexander Galán, y contarle que para nosotros es una tremenda alegría de poder tener presentes invitados internacionales de renombre en temáticas de biogeoquímica asociada a todos los sistemas marinos, que nos van a poder permitir comprender más las técnicas de muestras ambientales, cómo se procesan estas muestras, cómo se interpretan estos datos, con el fin de poder comprender cómo el cambio climático está impactando en todos nuestros ecosistemas, sobre todo los ecosistemas marinos o de aguas asociadas, por ejemplo, a ríos, lagos, que también es un interés que tiene nuestro centro, y en especial el doctor Galán. Comentarles que como institución nosotros tenemos una línea de desarrollo asociada a medio ambiente, y para nosotros es de suma relevancia poder contar con estas investigaciones, con esta vinculación internacional que permita no solo generar productividad, publicaciones o proyectos, sino también generar nuevo conocimiento y discusión, y al mismo tiempo poder apoyar a los tomadores de decisiones con datos científicos de cómo se debe intervenir y cómo se debe proteger nuestra biodiversidad, los sedimentos de nuestros mares o de nuestros suelos, etcétera. Y bueno, por supuesto, todos los otros ecosistemas marinos. Así que espero que disfruten esta jornada, que sea una experiencia de mucho aprendizaje, y que todas las interrogantes, las dudas o las ideas puedan emerger de manera colaborativa en este encuentro. Muchas gracias.
Okay, thanks by these emotive words, Dr. Maria Teresa, and after this brief contextualization, let's start with this workshop. So the main goal of this activity is to train young people, basically undergraduate and postgraduate students, young researchers and technical personnel in the areas of biological, marine and oceanographic science. And the training is in management of cutting-edge sampling techniques through molecular and isotopic tools for the analysis of the structure and function of microbial communities in extreme environments. So the first talk is entitled Following Geochemical Fingerprints to Understand the Biochemical Process of Organisms Living in Oxygen-Deficient Marine Environments, which will be presented by me. I am a marine biologist with a PhD in oceanography, and I am a CM researcher and assistant professor of the Catholic University of Maule, and I am also part of the Institute of Millennium Secos. My scientific experience is based on understanding the biochemical response of the microbial community in oxygen-deficient systems, water column and sediments also, and what is the response of these communities to environmental stressor, with a special focus in nitrogen, carbon, and sulfur cycles, and how the responses modulates the ecosystem function, and especially the stressor that has to be with the climate change. So before continuing, I will give you some general consideration for the activity, and I will be the moderator of the workshop. The talks will last 40 minutes plus 5 minutes for questions, which can be left in the chat of the transmission channel. We will try to solve as many questions as possible, but since time is short, I hope that the speakers will answer a question after in the chat if this time was not enough. Now then, if the time is not enough. In any case, we'll leave your emails of the speakers in case you want to make more consults after this activity. So I want to start with this image of the oxygen minimal zone of the Eastern South Pacific, which is the system where I have been developing part of my work in the last 18 years. This oxygen minimal zone is one of the shallowest and most intense oxygen minimal zones in the planet. It is distributed from the equator to around 88 degrees of latitude south, being more extensive and deep at 5 degrees south. In general terms, it is estimated that although these zones at a global level represent less than 1% of the total volume of the ocean, more than 50% of the nitrogen loss occur in these zones. This is a image courtesy by Ricardo de Paul, and you can see how is the distribution of this oxygen minimal zone in the Eastern South Pacific. And you can see that the biggest part of this mass of water is in the north part and close to the coast. And let's see what happened here. We can see The OMC developed in all eastern boundary system, you know, is a characteristic of all of these systems, as the Humboldt, California, Canarias, and Benguela system. And we can see here the global distribution of the main OMCs around the world, including the Arabian Sea. And in colors, you can see the concentration of oxygen around 400 meters. And you can see in this color, in the pink color, let me put the laser here. You can see the distribution of this OMC in the Pacific and the development in the Arabian Sea. That is really truly oxygen deficient conditions. And in Canarias, in the Benguela system, there is not so deficient, but definitely there is a development of these epoxy conditions. So all the eastern boundary systems are characterized by being affected by a boiling process. So briefly, the boiling process is a mechanism modulated by the wind that runs parallel to the coast toward the equator. But because of the rotation of the air, what is known as Coriolis effect, it generated a coastal ocean displacement of the surface water layers. Due to mass conservation, this volume of water moving offshore is replaced with deep waters. These waters are characterized to be poor in oxygen and rich in nutrients. So this injection of nutrients increase the productivity at the surface level, as you can see here as like a phytoplankton bloom. No? So this injection of nutrient-rich water from the depth, you know, moving to the upper part and put in contact with the sunlight, and this increase the primary production, and then this huge amount of material produced here in surface settle to intermediate waters, where it is remineralized, consuming the little oxygen that bring this already oxygen for mass of waters. I mean, the waters that is bringing with the boiling is already deficient in oxygen, but all of this material that coming from the upper part, you know, reduce in important form this the oxygen, so generating these environments truly deficient in oxygen. So here we can see how is the energy channel through the system, and we can see in the right part how the energy is mobilized from the, you know, big predators organisms, you know, when the oxygen is, or when the system is full in oxygen, to this condition when the condition is suboxic, you know, the energy is mobilized almost entirely by the microbial organisms. And you can see here at the left part how is the community. It's a complex community. So when the people talk about the dead zones, it's a really grown statement, because definitely there is a big community there. So they use definitely different compounds, and they use different compounds to oxidize the organic matter instead of the oxygen. So here you can see under oxygen deficient conditions, that is estimated around 2 and 4 micromoles of oxygen per liter. The microbial metabolism modulated by nitrate is activated, because this compound is the next most favorable thermal electron receptor to oxidize the organic matter. So the nitrogen dominate, the cycling of the nitrogen cycle dominate the system. So after the nitrogen, iron, manganese, sulfate, and organic compounds are the main electron receptors. But definitely what the oxygen is moving to really down conditions, this nitrogen metabolism is activated. So another important thing is to must consider it that the system is not necessary that the oxygen is gone completely to activate the use of NO3. So because this is generating much energy at the lower levels. So here we can see, for this reason, we will focus on the study of the nitrogen cycle in this environment, because definitely the nitrogen modulates most of the part of the energy, at least at the beginning of the oxygen deficiency. And this is a complex cycle that has compounds in many states of oxidations. Compounds can change from particulate to dissolved and even gaseous states, with each redox reaction involving at least one specialized enzyme, in some cases more than one. And you can see in red, all of these letters means different enzymes that modulate the reaction of reduction of oxidations. I want to highlight that under suboxygen active conditions, the nitrification process canonical denitrification process is activated, there is a mechanism by which NO3 is reduced through a sequence of reactions that end up in molecular nitrogen. This gas is so difficult to assimilate by the microbial community, and eventually this diffuses into the atmosphere. So that means that there is a reaction, produce this gas that eventually move out from the system, so they affect the productivity of the system. So molecular nitrogen is mainly assimilated by cyanobacteria. This is the only organism that is capable to do that. So definitely the other community is really limited by the production of these gases. However, depending on the levels of the oxygen deficiency, this reaction can stop at nitrous oxide, and nitrous oxide, this form of this compound. And this compound has the peculiarity that it's a greenhouse gas that is 300 times more radioactive than the CO2. That means that if you compare one molecule of nitrous oxide and one molecule of CO2, the nitrous oxide is more radioactive. The big difference is the concentration is really low compared between NO2 and CO2. And under these suboxic conditions, is also activated the metabolism of animals that contribute to the loss of nitrogen for the system. So from the perspective of the carbon, it is interesting to understand what triggers one or another mechanism, since while this canonical denitrification returns inorganic carbon to the system through the decomposition of organic matter, animal fixes carbon generating new organic material in this environment. That is, or that means heterotrophy versus autotrophy. No, I mean, the carbon is recycled, from organic to the inorganic part, and animals use this carbon, to produce new material in the system. So this is interesting if you see these both mechanisms from the carbon perspective. Thus, these new or these nitrogen loss processes or mechanisms limit the primary production because produce these gases that is not used for the community. And when produce this nitrous oxide, basically for incomplete denitrification, but I want to express that nitrification, that is an oxic process, that occurs maybe in the oxycline in the upper part of the system, is demanding oxygen, is a process that consumes oxygen for oxidated ammonium. But when the oxygen is really down, this ammonium is oxidated to NO2 and then is returned to the denitrification pathway. So nitrification under this anoxic condition or suboxic condition also participate in the production of this gas. So these conditions affecting the global climate because of definitely produce the gas. And the other thing is because they remove nitrogen from the system, regulate the function in general, and modulate the global biochemistry of the system. Okay, here I want to show this diagram. It's courtesy by Botandrup, this diagram that show how the distribution of the species, of the nitrogen species along the gradient of the oxygen minimal zone in the Eastern South Pacific. It can be observed that there is an important segregation in the process that dominate and in the accumulation of the different nitrogen compounds. No? And it is interesting to see that in the oxycline, for example, nitrification is the dominant process. As I told you, nitrification is a process that demands or that requires oxygen to oxidate the ammonium and then to oxidate the NO2 to NO3. But while in the upper part of the OMC, nitrous oxide is accumulated due to incomplete denitrification, but also due of incomplete nitrification in a process known as nitrifier denitrification. That is a process not very well known. And then when we move inside to the OMC, the nitrification and Anammox process dominate in the system. And it is interesting to see how while the nitrate and ammonium are consumed, you can see nitrate in blue and ammonium in red. There is an accumulation of nitrite that can show a decoupling between the process of nitrate reduction and nitrite reduction. So NO2 is an really important indicator, of the, not just of the condition of these mechanisms, but also it's important to see how is the condition in terms of the oxygen availability. and here Anamox in the core, but definitely there is a lot of control of this process in the upper part of the seed, because the organic matter is moving from the upper part, and then when they move to this oxygen minimal zone, because the difference in the density of the mass of water, the decay in the velocity of these particles is reduced, so a huge part of the organic matter is accumulated in this upper level. So this upper level is so interesting to see what happened, because all the substrate that trigger this mechanism is occurring there. Okay. And this consumption of nitrate inside to the oil seed allows me to talk about the first geochemical fingerprint. This is the main interest of this talk, is just to observe how is the system and how is the fingerprint that these mechanisms left there, to see how we can interpret that. So the first geochemical fingerprint that I want to mention is the nitrogen deficit, or N-star. This is a quasi-conservative tracer proposed by Gruber and Sarmiento, which is based on the deviation of the nitrite-phosphate ratio relative to the Redfield stoichiometry. In this figure, you can see the red line denotes the 16:1 ratio. This is the Redfield proportion in between nitrogen and phosphate. And the figure indicate that any deviation from this line to the left or to the right represent an excess of nitrate production or consumption relative to the phosphate availability that is expected to occur after the organic matter remyelinization. This geochemical fingerprint is represented by this formula, where the values represent constants included to adjust the average value of phosphate and NO3 found in the global at the global level. They use the authors use the distribution, the average distribution of these compounds to get the value to 0, and so they include these constants to get that. So this is the question, and in this diagram, or the value that deviate from the left to the red line, and that represent a favorable deviation toward phosphate. That means nitrogen is consuming while phosphate accumulate, represent a nitrogen deficit, which properly interpreted could represent nitrogen loss processes. In this diagram, this proposed by Capone and Knapp, it can be seen how the Redfield ratio between nitrogen and phosphorus is maintained when the organic matter is respired under oxide conditions. However, when respiration of this organic material occur in oxygen deficient zones, where the denitrification and Anamox processes are active, this relation is reduced. Now you can see here how this is a deviation. So here we can observe the temporal and vertical distribution of some nitrogen compounds on the continental shelf of Concepcion, Chile, that we measured during a year in 2009, 2010. You can see in the vertical the depth. This is a station that is over the shelf of Concepcion that has around 90 meters of depth. So you can see in the vertical the depth, and you can see in the x-axis the time is 1 year, January to January of sampling, and you can see NO3 in the upper part of this slide, and you can see in red how is the entrance of these nutrient-rich waters rich in nutrients, represented by this red one, this color here. And it's important to establish that the upwelling process here is modulated by seasonally. So during September, when the wind change from the south, this process is activated. So water coming from the seed is bringing to the shelf and bring these rich waters. So this activates the primary production, and you can see that definitely during the development of the upwelling season, you can see the accumulation of nitrite, especially here during the summer, but also here in the transitional periods. Ammonia is also accumulated there, and you can see nitrous oxide also accumulated, but not just in the bottom, but also in the oxycline. And here we can see the segregation. The origin of here is more related with the nitrification, but definitely here is more related with nitrification and complete nitrification. So The nitrogen deficits related to this end tracer become important during these events. So we can see here the accumulation of the values of this tracer is bigger in negative values in these transitional periods. No, you can see here in April, September, and during January, how is this value is get a really big deficit of nitrogen, and just in this part, you know, we can see that definitely is related with the nitrification and anamox process occurring there. So let me see what happened here. Okay, this is another important issue that must be taken into account when working in this oxygen deficient environment. The problem that is the problem associated with the measurement of the oxygen. You know, today the methods or instruments used regularly are not unable to measure oxygen concentration below 1 to 2 micromoles per liter. Profilers using oxygen sensors like the CTD that is shown here in the left part have a detection limit of around 20 micromoles per liter. And while the Winkler method achieves values close to the 1 micromole, it has the disadvantage that that is required samples to be taken discreetly and read in the laboratory. So you can see here the difference between both instruments that regularly are used and 1 micromole, that is the detection limit of the Winkler method, is a huge amount of oxygen for the microbial community that living in this part. However, Dr. Niels Peter group developed an electrochemical sensor that can determine ultra low oxygen concentration down to 1 nanomole per liter. That means 3 orders of magnitude lower than the traditional used methods. However, the associated cost to use that or to this instrument is really high, and it does not have the facilities to be deployed at the speed of the regular profiler at the CTD. So there are some considerations that we must to have when we use this kind of instruments. So with the use of this equipment that the Niels called stocks, Dr. Osvaldo Ulloa's group was able to determine that the core of the MC of the Eastern South Pacific present periods of anoxic conditions. Based on these measurements, Dr. Ulloa coined the term of anoxic marine zones and confirmed that under these anoxic conditions, as can be seen here in this diagram in blue, one of the main characteristics of this kind of system was the accumulation of significant amounts of NO2 that you can see here in red in this diagram. So in this, I will use this slide. This is one expedition developed by the Millennium Institute of Oceanography at the University of Concepcion along the Chilean coast and in a cross section of Iquique. You can see here along from the south to the north, and then a cross section here in front to Iquique, and that is possible to observe the distribution of the oxygen minimal zone of the Eastern South Pacific, how a more limited proportion of this mass of water. I mean, you can see the oxygen minimal zone here in this pink color. I don't know which color is this, but you can see all the area that occupy the oxygen minimal zone. But you can see that just one most small part of this water column is that we consider anoxic because the NO2 is accumulated. So nitrite accumulation is another geochemical fingerprint that can be used as a proxy of anoxic conditions, considering the limitation that we have to approximate to really get really low measurements of the oxygen. So it's a good indicator. It's another fingerprint I want to highlight. So using this data from the data from the same work published by Vargas et al., I would like to briefly talk about another geochemical fingerprint associated with this oxygen deficient environment. And this is one is based on the isotopic fractionation exerted by biological communities on the substrates they use with respect to the products that are generated. No? Through this approach, it is possible to identify the mechanisms that have occurred behind the isotopic fingerprint that remains in the environment on the principle that organisms prefer to use the light isotopes of the compounds. And this has to do with the behavior of isotopic molecules. The mechanism that makes isotopic molecules behave differently is known as isotopic fractionation, which is the change in isotopic composition that occurs when a compound passes from one state to another, or when it's transformed into another compound. And although this difference can occur due to chemical properties, it is the physical properties that mainly determine this fractionation. On the one hand, because heavy isotopic molecules have less mobility, and on the other hand, because they have stronger chemical bonds. Therefore, the energy required to transfer a heavy isotope molecule is greater, which means that the light isotope molecule in general reacts more easily. So for that reason, in general, the communities, or when the biological communities transform, these substrates use in general the light isotope. In this case, what we can observe here is that the isotopic composition of the delta 13 carbon in the dissolved inorganic carbon in the left panel, and in the particular organic carbon in the right side, for two different campaigns, present contrasting conditions. In 2015, a large isotopic fractionation between Dick and Pock was observed in waters of the anoxic marine zone, indicate that the predominance of in situ dark carbon fixation, while in 2018 this fractionation was lower, suggesting a predominance of a heterotrophic process. So this is a good fingerprint to identify which mechanisms are behind it. So using this isotopic fractionation information, these values, we're able to elucidate that during the campaign of 2015, sulfur-driven autotrophic denitrification greater than Anamox was the mechanism responsible for much of the carbon fixation inside the enzyme. And in this work, and based on the isotopic mass balance model, we were able to establish that up to 35% of the particulate organic carbon exported to the water was produced in this anoxic marine zone. So this is a really good approximation, to find that definitely inside of these zones, there is a lot of activity, chemotrophic activity. And the other interesting thing is we thought, previously that the animals were the main responsible for this. But definitely, based on this fractionation that we found, there is another mechanism behind of this fractionation. And in this case, is this sulfur activity, in these conditions, reduced sulfur compounds are accumulated, hydrogen sulfide mainly, and the communities use this hydrogen sulfide, reducing nitride. That means that combine sulfur and nitrogen cycle. And this denitrification in this case is not a heterotrophic process. This is important to have in mind. So in this case, this denitrification process is more autotrophic. So the communities can change from an state, a heterotrophic state to an autotrophic state. So in terms of the carbon balance, it's important, this change in the community. So now that we understand of the system is when a boiling process is occurring there, produce this organic matter, this rich organic matter that came from the surface to these oxygen minimal zones, and this rich material is consumed first in the way of these heterotrophic processes. But then, when the substrate is, produced, when inorganic substrate is produced, this community that are autotrophic found the ideal, conditions, to fix carbon. So these results are important for the carbon balance of these systems. It was established that these oxygen deficient environments are an important source of new carbon, increasingly the efficiency of the biological pumping and the sequestration of the carbon to the deep ocean. And finally, all the projections established that in the context of climate change, there will be an expansion and intensification of the oxygen marine zones, anoxic marine zones, like these documents. And this is occurring at a global level. And definitely this combined or together with the oxygenation process due to the increase in the water temperature and consequently to a lower dissolution of gas in the ocean, there are really good modellators of how much oxygen, the ocean is content in the future. Therefore, the use of these geochemical fingerprints will facilitate the approach to understand these problems. in the context of climate change. And definitely, this is my last slide. I don't know how is the time. Oh, maybe it's. And after this brief approximation regarding geochemical fingerprints associated with oxygen minimal zone, I give you space to the question, but not without first thanking for your attention. So I'm going to see what happened here in the transmission to see if there is some question. I don't know if there is someone here in so I want to see what happened here in the transmission to see if there is some question. I don't know if there is someone here in so I want to sorry. See what happened here. You can hear me. I can don't leave to share my screen. So la transmisión acá. Sí, there is some. So I cannot stop to share my screen. So yes, I need help. I cannot stop to share my screen.
Alex, maybe I can try to start sharing mine. Sometimes that helps. I'll just try that.
Okay, please, Bob.
Okay, to see if I can find the right. Just a second.
Yes, but I don't know if there is someone see in the chat of the transmission to see that there is question there.
It doesn't look. I don't see any questions right now. Okay, I'll try to share my screen.
Okay, good. Okay.
Is that working then?
Yes. No.
Okay.
Okay. Aparna said that she doesn't see any question there.
Alex, can I ask a question here?
Okay, good. Please.
I'd like to know if this phenomenon has happened before in the geological time. and how we understand that, because in the global geological history, there are cycles, right? That we have fossil and geological records. So, is there any evidence that this happened before and then was restored back to a more oxygenated levels of water?
Yes, definitely. This is not actual condition. This is historically we found the evidence that these oxygen marine zones was changed, in volume and in intensity. So there are people that working in these paleoceanographic, studies and determine which conditions was present here in the system, and modulate these dynamics of the ocean sea. And I guess our last speaker, Dr. Dharma Reyes, we will talk about that. Maybe she's more involved in these paleo studies. But definitely there is this. We in the record, in the sediment record, we can see how these conditions are changed and how it's related. For example, what these oxygen levels are related with the productivity of the system, for example, of the dynamics in the circulation. But definitely, Dharma maybe talk a little bit about this more.
Okay, thank you very much.
No, thank you. Sorry for the inconvenience because I couldn't see what happened with my presentation. But now we are on time, and I'm going to present the next speaker. To me, it's a real pleasure to introduce Dr. Botandro, who has supported my career for more than 10 years. Bo is a professor of microbiology at the University of Southern Denmark in Odense. His research focus is on biochemistry and microbial ecology, with special interest in the transformation of carbon, nitrogen, iron, manganese, and sulfur, and the influence of these processes on global element cycles. His major contributions include quantification of the role of microbial iron and manganese reduction in the aquatic sediments. He discovered of a new microbial metabolism, sulfur disproportionation, and he discovered also anammox or anaerobic ammonium oxidation by the anammox process. That is an important thing for fixing nitrogen in natural environments, as I showed you previously. And he has extensive investigation of the distribution and regulation of microbial nitrogen transformation in oxygen minimum zones. Current research topics include microbial nitrogen cycling in oxygen-depleted waters, sediments, and other environments, the interaction of carbon, sulfur, iron, and manganese cycling in freshwater sediments, anaerobic methane oxidation in the oxygen minimal zones, and biochemistry of adult range. So, welcome. Welcome to this activity, and please, you can share your screen now.
Thank you. Thank you, Alex, for this nice introduction. I just need to get this correct now. Should be here. Okay. Does it look like a presentation? It does, right? Thank you very much for the introduction and for the invitation to give this talk. Also, thank you to Alex for actually introducing much of the nitrogen cycle already, so I don't have to do that. I have some slides in the beginning, which I can go through quickly then. So what I would like to do today is to, it's a very methodological presentation I have today, where I would like to talk a bit about the methods we use to determine microbial nitrogen transformations in various systems through isotope tracing experiments, with special focus, because we are in this workshop, especially focused on extreme environments. I'm going to talk about the oxygen minimum zones that Alex already talked about, which are, at least in terms of oxygen, an extreme environment. So I'm starting out. I don't know if you want to show a picture of the nitrogen cycle, and it's just not just some arrows pointing all directions. I think at least this first image doesn't show the nitrogen cycle, but it does show some consequences of nitrogen cycling in the oceans, because it's a composite satellite climatography of chlorophyll A in surface waters. And as many of you probably know, algal or primary production in the oceans is limited to a large extent by nitrogen. So it actually also reflects the input of nitrogen to these waters. And you see, for instance, the waters, oops, sorry, how did that happen here? Just a second, I lost everything. Yes, I'll see if I can get a pointer over here, sorry. That the waters you know so well of South America, of Chile, are relatively chlorophyll rich due to the upwelling of nutrient-rich water, and the main nutrient here, of course, is nitrate that drives a lot of this. So nitrate is an important nutrient, but at the same time, nitrogen in nature occurs in a wide variety of oxidation states and wide variety of compounds. And when here I'm going to focus on the inorganic compound, there's of course also some interesting organic nitrogen chemistry, which I will leave out. But essentially, we find all these different oxidation states of nitrogen, from nitrate as the most oxidized to ammonium and amines as the most reduced form. Some of these are only found in small amounts, whereas others are really important, like, of course, N2, which is a major part of our atmosphere, and something like nitrite, which Alex just talked about, at least as a fingerprint of an oxygen minimum zones. So nitrogen has these two main biogeochemical significances. On one hand, it's a nutrient, which is incorporated into phytoplankton. We saw the stoichiometric relationship also in Alex's talk. And at the same time, it can be used in energy metabolism. For instance, nitrate, also nitrite, are electron acceptors for anaerobic respiration. We also just heard about that. And ammonium is a donor for lithotrophic oxidation in the nitrification processes. And there are more processes than that, as I'll get back to. But it means we have a redox cycle at the same time as we have a cycle of assimilation, dissimilation cycle into and out of organic matter. Other interesting aspects, which Alex also mentioned, is that N2O, laughing gas, is a very strong greenhouse gas. It's also the major ozone destroyer these days. And other relevant aspects of nitrogen biogeochemistry would be that both nitrate and nitrite can buffer in some systems for hydrogen sulfide. That's also the case in oxygen minimum zones and in sediments, and maybe they also to some extent buffer from methane emissions for some systems. So the nitrogen cycle has its sort of built-in paradox in the fact that there's so much nitrogen in the atmosphere. So here about 4 × 10⁻21 grams of nitrogen in the atmosphere, and still we have nitrogen limitation in many systems. And this is, of course, because of this triple bond in nitrogen, which is hard to break, has a high activation energy for breaking, and makes it such that most organisms cannot utilize into in the form it is in the atmosphere. Comparably, the pool of reactive or bioavailable or fixed nitrogen, whatever you like to call it, is more than 1,000 times smaller than that. So these are forms like ammonium and nitrate and organic nitrogen and so on. And these two pools are then connected, as we also already heard, by first of all, nitrogen fixation, which in the old days was a microbial process. Now it's also an industrial process, but still microbial nitrogen fixation is a major source of fixed nitrogen. And if that was all that happened, there would be no nitrogen limitation on Earth, because then those organisms could fix as much nitrogen as we needed, or as organisms needed, and then that would be fine. But at the same time, because nitrogen is also a respiratory substrate, it's delivered back to the atmosphere by a process or more processes. We can now call them microbial denitrification. As in a few minutes, I'll be a little bit more detailed about what processes these actually are. It's not just one type of process, but it's a few different kinds of processes. So this is important. This balance between fixation and denitrification is important then, because nitrogen often limits primary productivity. And if we look, for instance, in an oceanic reactive nitrogen budget, or a compilation of different budgets, it would look something like this. Inputs and sinks of reactive nitrogen, fixed nitrogen in the oceans. We see that there is riverine inputs from land, there's atmospheric inputs, there is nitrogen fixation on one hand, and then the major sinks we're looking at are denitrification. Again, we have denitrification in sediments, which is in this case about 2/3 of the sink term, and denitrification in the water column, which we also just heard about, in oxygen minimum zones, which is about 1/3 of the sink terms, and then a little bit of burial also over here. But what you also see is that there are huge error bars on these numbers, first of all, which means that they're not that well determined. And at the same time, if we compare these denitrification rates to the total pool, the standing stock of nitrogen, fixed nitrogen in the oceans, we see that we have a turnover time of oceanic nitrate, which is only on the scale of a couple of thousand years, something like that. So this means that small changes in these denitrification processes could actually heavily impact the availability of nitrogen in the oceans, which would then again impact primary productivity, which would impact the drawdown of CO2, the carbon cycle, and so on. So it's really important to understand these processes, to quantify the processes, and to figure out how they are controlled. For instance, just for understanding the distribution of chlorophyll that I've showed again in this little graph down below. So to do this, we need to take a closer look at the nitrogen cycle, and Alex already did show a more complex nitrogen cycle than what I was just indicating before. Here's another version, essentially showing the same thing. We have to the left assimilatory uptake of nitrogen into organic matter, the release through mineralization again into ammonium. And here we've split it up so that there's both an anoxic environment here and an oxic environment, because the downstream processes, or the processes that can then deal with ammonium in these systems, are very different. So if we're under aerobic conditions, as we already heard, we have nitrification producing nitrite, producing nitrate, and these could be assimilated back again. This is not a problem. We still haven't lost any nitrogen from the system. But if these compounds then get into anoxic environments, they can be consumed by the denitrification process, by the Anamox process, which Alex also mentioned, which is this co-proportionation of nitrite and ammonium forming N2. It actually also forms a little bit of nitrate through the assimilatory, the carbon assimilation, the CO2 reduction is coupled to nitrite oxidation. And in addition to that, there are other potential sinks for nitrite. You see, nitrite is really here sort of a pivot in the whole cycle. There are many arrows leading into and going out from nitrite. But nitrite could also be reduced to ammonium through a process which we call desimilatory nitrate reduction to ammonia. Or it could be, as we also know now, it could be denitrified in a different pathway known as methane denitrification, which has different intermediates, for instance, not N2O as an intermediate. So it's a complex cycle, and we need to understand all these processes which often are co-occurring at the same time. And how can we do that? Well, the best way, I would say, and the way that I've been using for a while, and that I'll be talking about today then, is the use of 15N as a tracer. So 15N is the rare isotope, stable isotope of nitrogen. It's about 0.4% of the nitrogen in natural abundances, and then which means that if we can use 15N as a substrate, and we have a mass spec, an isotope ratio mass spec like this one, for instance, to analyze the substances, then we can actually, at a very high sensitivity, detect these different kinds of transformations. And this is a big advantage. In the oxygen minimum zones, for instance, typical rates of nitrogen transformations are in the range of 1 or even actually less than 1 to maybe when it's really, really active to about 100 nanomolar per day. And if you compare that to a background concentration of nitrate of maybe 20 micromolar, you see that you have to look at very, very small changes in a very large pool if you did not have these isotope methods to use. Isotope methods have been used for a long time, and I think this paper by Goring and Dugdale from '66 is maybe the first one where they used 15N nitrate to detect denitrification in a water column, in this case from a subarctic lake, but Goring continued this work and has also worked in upstream minimum zones actually. You could also, as I showed you before, the nitrogen cycle is relatively complex, and there are lots of intermediates, and some of those intermediates one could measure directly, and maybe also measure changes in the intermediates directly, which has been used previously in different types of inhibitor treatments. For instance, a classic way of measuring denitrification was to use acetylene as an inhibitor, and then look at the accumulation of N2O, which you can do with very high sensitivity. But again, I would say that 15N incubations have an advantage against compared to this, because first of all, you can measure different, look at different processes simultaneously, but also you don't have these side effects that inhibitors typically have. Acetylene doesn't only inhibit denitrification, it also inhibits nitrification, which is an issue. And there are many of issues associated with the use of inhibitors. They're still useful, but they have their limitations. And this is another advantage of 15N. And finally, which I think is really important to note, is especially if we're interested in the production of N2 or N2O, which are these important gases, as we heard about already, 15N or isotope tracing has a special advantage that we can look at the pairing of isotopes coming into these compounds. And this is when Alex was showing rates of NMOX denitrification and other processes before, especially of NMOX. This is basically based on some of these principles of isotope pairing into these substrates. So just to illustrate what I mean here, a very simple little graphic. Here we have a denitrifying microbe. It is supplied with some 15N nitrite and will then, of course, produce N2, which is double labeled. Here we have 15N, 15N over here. If we have 14N nitrite, it'll produce double labeled N2, which is 14N, 14N, or mass 48, or 28 here, mass 30 over here. If, on the other hand, we have an NMOX bacterium, and we again supply this NMOX bacterium with 15 nitrite, what we see is that it will produce single labeled N2, 14N, 15N, mass 29, because it's combining the nitrite with unlabeled ammonium. And similarly, if we label the ammonium, in this case, it will also produce this mixed product. So we can actually, for instance, in an experiment with 15 nitrite, we can actually distinguish whether N2 is produced by one or the other. And as an example of that, here's the first study I was involved in with NMOX, which was in sediments. We have a little detour to sediments here, but I'll get back to water columns again later. And we did experiments with marine sediments, and we added different combinations of tracers. First of all, we added 15N labeled ammonium to the sediment. And when we did that, we saw there was a production of this single labeled N2, 14N, 15N, whereas there was a tiny little production of double labeled N2 down here, but not very much. Then if we, so this, this of course indicates that the ammonium supplies one N and that the nitrate supplies another N here. If we switch the compounds around, again, we see there's a production of the double labeled, the single labeled N2, but now we also have a production of double labeled N2. So how do we explain this? Well, the single labeled N2 again is produced by a combination of ammonium and nitrate, whereas the double labeled is denitrification. So the production of double labeled N2 from two nitrate molecules. So actually in this incubation here, number 2, we can actually measure both the NMOX process producing the single label and the denitrification process producing the double label. And if we want, we could go in and label both of the compounds, and then we can see that we get the largest production of double labeled N2 now forming from both denitrification and from NMOX. And there's a little bit production of single labeled N2 down here, which is explained by the combination of unlabeled ammonium, which is always present in the sediment, with 15N labeled nitrate. So it's a relatively powerful tool to determine the relative importance of these processes. And the first time we used it was just in a few sediments, when here you see results just focus on the columns here, where we see that at 3 different sediments, different water depths, from shallow base, Aarhus Bay at 20 meters to deep Skagerrak at 700 meters, we see very different contributions of NMOX, or anaerobic ammonium oxidation, to N2 production, going from about 60–70% at the deep station to only a few percent at the shallow station. And people have done more of these types of incubations and compiled something like this, a compilation of the relative importance of NMOX for many different kinds of sediments at different water depths. And what we generally see is that in shallow sediments, the contribution is relatively small, whereas we get into deeper waters, the contribution is bigger. And very, very briefly, because we're not going to talk so much about sediments here, but very briefly, this has to do with the competition between NMOX and denitrification for nitrite, which both, it's the primary substrate for NMOX, and it's also, of course, a substrate for denitrification. I should also mention here that this isotope pairing has also a very interesting use in whole core incubations of sediments. So here, a very simple illustration of a sediment core. And if we add nitrate, or we haven't added it yet, in the sediment here, we have, assume we have some nitrate in the overlying water, and we have a production of nitrate in the sediment due to nitrification, ammonium diffusing up from below. There will be denitrification going on. Without any labeled adlers, the denitrification will produce unlabeled N2, 14N or mass 28. But now if we add 15N label to the water, the 15N will diffuse into the sediment. And now we will, in the zone of denitrification over here, we will have both the presence of 14N and 15N labeled nitrate, and we will generate all 3 different masses of N2. And if this is all that's going on, we can actually, from the relative production of the different masses here, we can actually calculate a denitrification rate, which will depend on the degree of labeling, or Q, here in the sediment, in the denitrification zone. So by this random isotope pairing, which is what occurs during denitrification, we have the relative frequency of the 3 masses that form are these this time binomial distribution, where 30N2 forms at a frequency of, it's the square of the relative abundance of 15N nitrate, and the 2 others then form at these other formulas. These are the only equations I'm going to show, I think, in this presentation. So don't be scared if you don't like them at this stage. So we just heard about this. This works well if we have NMOX, if we have denitrification in the sediment. But I just said that we also know now that in many sediments, NMOX is also an important process. We have ammonium coming up from below, which will also produce, in the case of we have added 15N nitrate, will produce both single labeled and unlabeled N2. And actually we cannot now, because of this, we cannot distinguish the relative importance of these. But there are still options here, because if we measure the isotopic composition of N2O, in this case, we can actually, by that, determine what the frequency of the relabeling fraction of nitrate is, because N2O is only produced by denitrification. And then if we compare the relative abundances of produced N2O to the abundances of produced N2, we can actually go in and calculate the total denitrification and NMOX in the system still. So it's pretty smart and really shows the strength of the stable isotope methods. But let me switch back now to looking at water columns and a little bit more in detail about experiments. So how do we actually do this? Well, here's a graphic. It was not made to illustrate nitrogen transformation. The graphic was actually made to illustrate how we do incubations to measure anaerobic methane oxidation in these waters, but it doesn't matter. The principles are the same. And we have these different steps. So typically we would be sampling water from a water sample like a Niskin bottle that you see here. We would let the water go into a bottle of some kind, fill the bottle with overflow to ensure if we're sampling from anoxic depths that we flush out as much of the air and the oxygen as possible. Then we can create a headspace, and we could add the 15N tracer at this stage. And then what we usually do, because now we know, okay, the water has been contaminated with oxygen, we know that we need to get rid of that oxygen again. So we would purge the water with helium. And we use helium instead of nitrogen because of the mass spectrometry we want to do, where we want to measure nitrogen. We also use helium instead of argon because argon often contains relatively large amounts of isotopically heavy nitrogen, which can interfere with the measurements. Next step then is that we dispense into some smaller vials. In this case, we're using what we call excitaners here, small vials, and we can do make a lot of these. And then the next step is that we will insert a headspace, and we'll flush this headspace because we still need to get rid of oxygen, because even just by dispensing the water again, we have introduced oxygen into the system. We want to get rid of that. We don't want to inject labeled methane in this case, because this is not a methane oxidation experiment, but we could do that. We could inject other things here, inhibitors or anything else. We mix, and then we incubate these vials for a while. Finally, we kill them. We terminate the processes by adding zinc chloride or mercuric chloride in this case, and we take them to the mass spec, and then we start all the calculations. So this is the basic outline that we're using, and that other people have been using for a long time to measure water column processes. Basically building on the method that was first published by Terje Dalsgaard and others in 2003, where they went and measured denitrification and NMOX in the water column of the little Golfo Dulce, which is in Chile, no, not in Chile, sorry, in Costa Rica, a small embayment, anoxic embayment, which has conditions similar to oxygen minimum zones. And here, this is actually the first quantification of NMOX in an anoxic water column. Sorry. A lot of compilation, a lot of measurements have then since been made, and this is a compilation I made in 2012. After that, there's even more work been done, but just the general picture that emerged from all these measurements about the relative importance of NMOX and denitrification in oxygen minimum zones was, at least to begin with, that in OMZs or oxygen minimum zones, which are indicated by triangles here, very often all the N2 production that we could measure was due to NMOX. The blue circles here are from Golfo Dulce. This is the data I showed over here also, but at much higher rates. And here we see denitrification becoming more and more important. And then finally, when we get up to very high rates, as we, for instance, found them in some anoxic fjords, it's completely dominated by denitrification. This was a picture that existed for a long time, but now it's getting more detail, and we have slowly figured out it's not as simple as this. I'm going to show you some new data, which we haven't published yet. It's produced by my great colleague, Laura Bristow, and with some assistance of myself, from the Eastern Tropical North Pacific oxygen minimum zone. Again, you've seen before oxygen profiles like this, where oxygen goes away at depths of something between 50 and 120 meters. In this case, the accumulation of nitrite as a good fingerprint of this anoxia here. And here, in incubations with 15N labeled nitrite, we were able to measure both NMOX denitrification, nitride oxidation. In principle, we would also be able to measure DNRA, but there was no detectable DNRA in these systems. But all this can be done. And again, it shows the strength of these types of incubations. And here we see very interestingly, actually, a sort of classical ammonia NMOX profile where the rates peak close to the oxygen-anoxic interface as nitride accumulates. We have high rates of NMOX here, and then they decrease with depth into the oxygen minimum zone. On the other hand, denitrification only picks up in deeper waters. There's not much going on in the shallower waters, and then the rates pick up. And I would say when I say pick up, they don't reach extremely high rates. These are actually still very, very low rates. And I think this is actually one of the reasons that it wasn't accounted for before, that we thought NMOX was the only process of importance here, but simply because we've improved the sensitivity of the method so much now. We're measuring at a detection limit. The stipple line here actually indicates our estimate of a detection limit for denitrification. And as you can see, we are below or close to, slightly below actually 0.1 nanomolar N2 per day, which is a very low rate. So we're learning a lot about these processes based on these types of incubations. So it's all relatively easy, right? It sounds like you go out, you put the water in your bubble, you do all this. Okay, there's quite a few steps involved, but still it's relatively easy. Are there no problems in these types of incubations? Of course there are. And the greatest problem we probably have is oxygen contamination. I already hinted a little bit at that. There's something called bottle effects. There's something called concerns about aggregates in the water column and the effects they might have. And there are also some considerations about the enrichment we're doing while adding the tracer to the system. And you might sometimes feel a little bit like the person on the important or well-known painting here. So going back again and thinking a little bit about these steps, now it's a simplified graph compared to the ones I showed you before. What do we know about oxygen contamination associated with this, with sampling here? Well, first of all, we know for sure that if you sample in a Niskin bottle, you will contaminate the the water with oxygen at concentrations of somewhere between a few hundred nanomolar to several micromolar. I would say, as a rule of thumb, there's probably like a micromolar of oxygen in the water once it comes up in the Niskin bottle, even if you sample at a depth where it's anoxic. And that's because the plastic contains oxygen. The PVC that these bottles are made of contains oxygen. It diffuses into the water. You can get better if you, instead of using Niskin bottles or GoFlow bottles, use a pump car system where it's all pumped. The water is pumped directly from depth onto deck. And if you pump for a while, the oxygen in the tubing will go away, and you can then get down, this is measurements we made, to something like 30 nanomolar is the best we've done, and up to maybe half a micromolar. But there's oxygen there. Okay, then we can go on, degas it, as I showed you before, and bring down oxygen again to very low levels. We can dispense, and if we're very, very careful and do it in an anoxic atmosphere, in a glove bag, we can maybe keep the anoxic conditions. But if we don't, we might again contaminate the water with oxygen. And then finally, when we have it in the excitaners, it turns out quite often, as you see here, that oxygen concentrations will increase in these excitaners, although the lids are supposed to be gas tight. So why does all this happen? Well, first of all, I already said it, plastics contain oxygen. Here are some numbers on the solubility of oxygen in different types of plastic. And what you see is that many types of plastic, oxygen is more soluble than in water. The blue line here indicates the solubility in seawater. So if you're using Teflon, if you're using polycarbonate, or if you're using acrylic or plexiglass, you will have more oxygen in the plastic than you have in the water. That's worth remembering. Also down here, note that if you're using butyl rubber, which is what is in typical stoppers in the vials we use for incubations, there's also a lot of oxygen in there. And this is the problem when you do incubations. You can be very careful about removing the oxygen, but then you add this stopper or this septum, which is butyl rubber, and it's full of oxygen, and this oxygen is going to get into your samples. unless you do something to protect it. To the right here, you see a table of different measurements, and the only ones that look really good, you can see we are measuring some hundreds of nanomolar, these are nanomolar concentrations of oxygen in excitators. The only time it really works is when we're using deoxygenated septa. So it's important that the septa we use are stored under either nitrogen or helium, or at least an anoxic atmosphere before the use. And this has to happen even months before you start working with these things. Here are some very nice continuous measurements of oxygen in different types of vials. Up here you see what happens if you add anoxic water to an excitator and the septum was not degassed and there was no headspace. The oxygen concentration in the excitator is increasing rapidly and gets up to several micromolar in this case. Whereas if you have degassed the septum, at least you're good for a couple of days before the oxygen from the outside breaks through the septum and starts an increase in the water here. Here are some similar examples from serum bottles. Of course, it doesn't really matter what kind of bottle you're using. If you're using a butyl rubber septum, you need to be careful. And then the question, of course, to ask is, what harm does this do? Is this important in any way? Why do we care? And we do care a lot, as you see here, different examples of the measurements of the sensitivity of different processes to oxygen. Here we have some really nice measurements by Laura Bristow again, measuring rates of ammonium oxidation, nitrite oxidation as a function of oxygen concentration in manipulation experiments. And what you see, especially in these magnified inserts here, that even oxygen concentrations of a few hundred nanomolar are enough for these processes to occur. And Alex also was talking about this. At the same time, if we look at how does oxygen inhibit denitrification or anammox, we can see that, well, anammox actually continues in this case in many systems up to concentrations of 5 to 10 micromolar, or sometimes even higher. And denitrification also is not inhibited immediately by very low amounts of oxygen, but maybe has a half saturation, half inhibition concentration of about 200 nanomolar. So clearly these processes can overlap. And if we add oxygen to the vials, which shouldn't be there, then we will affect the outcome. So what are the recommendations here? Well, avoid plastics and elastomers, rubber, as much as you could. And if you need to use it, it has to be degassed. It has to be put in an anoxic atmosphere, oxygen-free atmosphere before use. And that takes weeks, if not months. Use glass. In other systems, people are concerned about glass because of trace metals coming from the glass. It doesn't seem that trace metals play a big role in oxygen minimum zoned waters, so we can do that here. And then it's really important that you just don't assume that there's no oxygen in your incubations, but actually try to measure it in there if you have the equipment for that. And these days, optical oxygen sensors are catching on. Alex showed the other very highly sensitive stock sensors, which are also great, but there are also other options with optodes. Still, we know that whatever we do on deck has been exposed to oxygen. We cannot be totally sure that they're not irreversible effects. So we actually still need to think about ways of doing incubations completely without oxygen contamination. And I think this means incubations that are done in situ. A little bit about the other term I mentioned before, namely bottle effects. So what is a bottle effect? Well, that's sort of a term we use when we don't really know what's going on, but it's a well-known fact that if you do incubations in glass bottles, or in bottles in general, it doesn't have to be glass, and let them run for a while, all of a sudden things go crazy. All of a sudden rates increase immensely. The reasons for this, I don't know. I've suggested some here, but we actually don't know why that is. But at least it's a fact that it happens, and we see it often, not always. But if we do longer term incubation, so you need to keep your incubation short. You might also need to do time courses to check that there isn't something going on. that you don't see an acceleration of rates, which could be due to a bottle effect. And usually we would never do incubations longer than 24 hours, actually, because in experience, in most systems, the bottle effect doesn't kick in until after 24 hours. There are some examples over here, actually from waters off Iquique, where you see after 36 hours, all of a sudden, the nitrite concentration increases strongly, indicating that we have some acceleration of nitrate reduction, which is way beyond what is realistic as an in situ process. Finally, just a little bit here about the role of aggregates. There's something wrong with this slide here. It looks a little bit bad, but I showed you before this very variable oxygen sensitivity of anammox. We have measurements from different places, different locations of Peru. Some are offshore, the blue ones are offshore, the red ones are inshore. And what we see is that the oxygen sensitivity of anammox is quite different at these sites. It looks like the offshore stations have higher sensitivity than the inshore stations. And it could be due to aggregates. That was at least hypothesized here, that in coastal waters you have more aggregates. Aggregates have anoxic microneches, and that could be part of it. So it's something to think about in general when you do incubations. Is there a role of aggregates? Another example here from the Eastern Tropical North Pacific shows how the presence or absence of particles affect rates. So water was incubated in bulk or after filtration in 1.6 micron filters. And you can see the rates here up here in the upper panel, where we see that the gray rates are the bulk rates and the blue rates are those in the filtered water. And what we see is that especially for processes like nitrate reduction, denitrification, there's very little activity after filtration. Actually, there's no detectable activity here after filtration. Whereas anammox and ammonium oxidation, nitrite oxidation are not as affected by this. And the idea here is that especially these processes, nitrate reduction, denitrification, which are organotrophic, are associated to the particles. This is also supported by this general particle support is supported by the transcripts of these functional genes that you see down here. The last thing I want to remember to mention about these different methods here, of course, another thing to think about is what are we doing when we add the tracer? Are we changing the concentration so much that we are actually not measuring real rates, in situ rates, but how are rates affected by concentrations? And this is a concern, especially for ammonium, because ammonium concentrations, as we also heard before, can be very, very low in oxygen minimum zone waters. And if we do these tracer experiments, we typically add maybe a micromolar or something like that. And that is a real concern. In that case, you probably would need to say that you're looking at potential rates. But for anammox, you have the advantage that you can do experiments where you add ammonium, or you can do experiments where you add nitrite. And you can compare these and then get an idea of the in situ rates. You can, of course, also, if you have the energy and time, you can actually do your own kinetic experiments. And here's examples again that Laura and I did some time back of kinetics of anammox relative, dependent on the ammonium concentration, on the nitrite concentration, and also on denitrification on nitrite concentration. And we generally find very low Km values, but this is something that we need to consider, and everybody should consider when they do these kinds of incubations. How are we changing the concentrations, and how would this affect the results we're getting? So finally, as my last slide, I would get back to where we are right now, some of the news things we've started doing. And this, in this case, is about N2O production. We heard about N2O. It's this powerful greenhouse gas, and it's important to figure out why is N2O accumulating in these, usually around the boundaries of these oxygen deficient waters. Alex also showed an example of that. Is it due to N2O production from nitrification, which we see here, or is it, which can be either through this hydroxylamine oxidation pathway, it could be through nitrifier denitrification, or is it actually associated to heterotrophic denitrification? And again, we can use the isotopes as useful tracers to figure this out. And what we're working out now is actually using not only 15N, but also 18O for this. So if we add 15N labeled nitrite, for instance, we should see 15N labeled N2, and we can look at the isotope pairing in the N2O and see if it comes from this pathway, or there are other pathways that combine into the N2O here. But now we have the opportunity, because N2O is not only N, it's also O. So we can actually also use 18O labeled oxygen. And 18O labeled oxygen would be incorporated via hydroxylamine. The ammonium monooxygenase produces hydroxylamine, which is the source of N2O in one of the known pathways, and would then form 18O labeled N2O. You could say, but wouldn't also 18O come the other way around? You could produce nitrite, you produce NO, you produce N2O. And it would eventually, but actually it turns out that due to isotopic exchange, maybe also a lot of this, first of all, the oxygen is diluted into the big nitrite pool here, but there could also be exchange here where it's actually lost from the system again. So it doesn't get down the other way here. This, of course, requires, if we really want to do isotope labeling experiments, it requires a special mass spectrometer, because now we can actually form 5 different masses of N2O, the completely unlabeled here to the completely labeled over here. And this is what we're trying now. We have a mass spectrometer which can do this, and we're trying to see if we can make that work out. But I don't have any, I don't have time, and I actually also don't have any results to show you yet. So that will be just a little teaser here. So in summary, 15N incubations are a powerful tool for determination of nitrogen transformation rates. I hope I've convinced you about that, and can provide, isotope pairing provides unique insights that you can't really get in other ways, especially when we're looking at N2 and N2O production, of course. And whenever you go and do these types of experiments, there are some important considerations that you need to make. Control of oxygen concentrations, monitoring of oxygen concentrations if you can. Think about incubation times, think about substrate kinetics and amendments and so on. Thank you for your listening, and I'm happy to take any questions.
Thank you all for this interesting presentation. This is clarifying to me, and especially for the people that working with these methodologies. There is not a question in the chat, but I have a couple ones, and I want to know, for example, is you show this vertical segregation, you know, in the water column between Anamox and the nitrification, and even between NO2 oxidation and the nitrification.
Yes.
The nitrification is more related to the core. Anamox is more in the upper part.
Yes.
And we saw the same distribution here in the OMC of Iquique in the north part. You know that system. You know why this is this segregation? We have many, you know, suppose that is dependent on the organic matter that coming from to the surface, but I don't know if you check more in detail that.
Well, of course, so far it's because this is relatively new data also. So it is basically an observation, but we have the hypothesis would be that well, Anamox depends on ammonium. Ammonium comes from degradation of organic matter, so you would expect the highest rates of organic matter shallower as the organic matter rains down and is gradually consumed in the oxygen minimum zone. So that all makes sense. What's more surprising is that the denitrification then peaks deeper in the water column. But I think that the main reason for that is that in the shallow waters, nitrate is not reduced all the way to N2. It's reduced to nitrite. So nitrate reduction to nitrite is a really important process in the shallower waters and can occur at relatively high rates. but only in the deeper waters when for some reason maybe the nitrite concentration gets high enough and stimulates denitrification down there. So you see the accumulation of nitrite, of course, into the core. And maybe there could be other factors also that we don't know. But it's mainly the absence of denitrification in the shallower part of the OMC, which is interesting, and I think that's due to nitrate reduction. That organic matter oxidation is coupled to the reduction of nitrate to nitrite.
So, and the other question is related with that, and is about this NO2 oxidation that Laura Bristow found in this oxygen minimal zone. So that means that this step of the nitrification is not controlled by the oxygen. I mean, is less controlled than the ammonium oxidation?
Well, this is a big question, and it's something others have observed too. It's not the only place it's been observed here, but definitely and that's Laura's project, so I shouldn't say too much about it. It's what she's looking into, but it's definitely an observation that we have this nitrite oxidation, reoxidation of nitrite in the upper part, at least, as you also saw here, of the oxygen minimum zone. And it's a big question, how can that happen if there's no oxygen? It actually shouldn't happen. Nitrite oxidation is an aerobic process. And people have been speculating about other explanations, other electron acceptors, but there are no real other electron acceptors. So another option is that it's actually nitrite dismutation. People have been talking about that, which is energetically favorable and could be part of it. But at present, I don't think we have a good explanation for why this is happening. But it seems to be important there, and that means that we have a lot of cycling of nitrogen going on in the upper part.
I have more, but the last one I want to ask here is, did you mention something about the argon limitation to use in this kind of experiments? What is this limitation?
Well, the problem that I've run into, and then I just stopped using argon, so maybe it's not a problem all the time, is that apparently when argon is produced, at least sometimes you get relatively high concentrations of 30N2 and 29N2, but especially 30N2 in there. I guess it's just from the way it's produced, and gives you a very high background of 30N2 and 29N2. So the compounds that you're actually going to look for in your samples. And that disturbs the measurements quite a bit. I've also seen some mass specs behaving strangely with argon samples. I don't know more about that, but that's just an observation. But it's a bit unfortunate because argon is much more accessible than helium, but it looks like we're stuck with helium for a while at least.
And maybe cheaper. Okay, well, thank you a lot for your time and for doing part of this activity. And
you're welcome.
And we will see again soon.
Yes.
with more experiments to process. Thank you all.
You're welcome.
So the next speaker is my colleague, Dr. Aparna Banerjee. She is an investigator of the CIAM and assistant professor of the Catholic University of Maule. She has interest on applied microbiology and microbiology of extremophiles, focus on the extreme temperatures of the springs, Antarctic ecosystem, fumaroles, etc., and different bioactive polymers produced by them, majorly polysaccharides. She's looking into using them as food additive for food industry in a funded seed and INACH regular project, and as biomaterials for nanomaterials preparation for various biotechnological applications. Apart from that, she also has interest on microbial genomics and proteomics. To me, it's a real pleasure to introduce Aparna, and Aparna, please, the public is yours.
Thank you, Alex, for the nice presentation of me. Am I audible? I'm going to the presentation mode. Is the sound fine?
Yes, it's fine.
Okay. I guess it's full screen now, isn't it?
Yes.
Please.
Okay. So hello, everyone. A very good afternoon or a very good morning, wherever you are in the world. I'm Aparna Banerjee, and presently I'm working as assistant professor in SEAM, Universidad Católica del Maule, Chile, together with my colleague Alex, who is organizing this seminar today in the course of vinculation project by UNIT. So today I'm going to talk on sampling and isolation of thermophilic bacteria from hot spring environments for its bioactive compounds. Mainly, I will focus on three parts: among extreme environment hot springs, then the thermophiles and its bioactive compounds, and the biomaterials, these bioactive compounds that they are producing, their biotechnological applications. So even after after my talk, if you have any questions, you can write back to me. So I have shared there my mail ID. So to start with, I will talk a little about that microbes are omnipresent. Normally, when we think about microbes, the general idea is they're in water, they're in air, or they're in terrestrial or aquatic ecosystem. But other than that, in all possible inhospitable environment, they are there. In Chile, we are naturally gifted by the nature with a lot of extreme environment that can be possibly thought. For example, we have permafrost regions, we have fumaroles, we have hot springs, we have soda lakes, many of them. But in all over the world, there are different extreme environments where microbes are omnipresent, and in different environment, the strategy of microorganism is different to survive. So today I'm going to focus mostly on hot springs and how we are cultivating them, how we are getting the different bioactive compounds, and what are their applications. So to start with, when we talk about extremophile, now another term has came, that is polyextremophile. When we think about extremophile, it's like the organism that grows into an extreme factor, that have the capacity to grow into an extreme environment. For example, if we consider hot spring, the nutrient concentration is low, species diversity is unknown, temperature is high, oxygen availability is low. But when we think about poly-extreme environment, the factor poly is the environment have more than one extreme factor for the microorganism to survive. Can be abundant of heavy metals, extreme temperature, solar UV radiation. So I'm going to present the results of both the environment among some of the environment that I have worked, to give a little comparison between the environment that I have worked in India and what we are working in Chile. So in India, the hot spring that we were working with, that has among extreme different abundant heavy metals, we had high arsenic content and the temperature was high. But in case of Chile, the extremity is more in compared to India, because the hot springs that we are working with, normally in Chile, they are volcanic in origin. So there are different trace metals abundantly present. The temperature is high. There are abundant solar UV radiation, and often the pH is also a little extreme if it's a hot spring or it's a fumarole. So in different time period, there have been different rudimentary works on extreme environment, and that have changed the concept of microbiology, that have changed the concept of sampling, on getting the bioactive compounds. So back in 1970s, there was the first work on Yellowstone National Park, the hot spring and geyser of Yellowstone National Park. Now it's very known, but that was the first time that this work have been done on Thermus aquaticus, and that we are till now using Taq polymerase. So back 50 years back, that exploration has started, that has paved the way slowly in years after years. Then in 2003, after almost 30 years, there was a work in Marx-like soils of Atacama in Chile. That was also the work of Yellowstone National Park, that focused on one extreme environment, that is extreme temperature. And then in case of Atacama Desert in Chile, that focused into the dry limit of microbial life. So in different years now, a lot of people are working on Atacama, but the one very fast work that proved that Atacama soil has all the Mars-like feature was back in 2003. And then there is a recent work in 2020 on Mexican Lake improbable oasis. That's also another beautiful work on extreme environment and microorganism. So how these microorganisms adapt to the fitness there? Because most of the microorganisms that are there in those extreme environments are non-cultivable. And among the 1% culturable microorganism that we get, it's often difficult to cultivate them. So how they adapt to the fitness? They often have some loss of function mutation. The stability of the protein is different. They produce different stress response enzymes. The membrane fluidity is different. And one common factor is often they produce biofilm. So the mat that often we see in different hot springs or different thermophilic environment, that is basically the biofilm network that the microorganisms are forming for years after years. And more thick the biofilm network is, more undisturbed the environment is. So we're going to talk a little more about biofilm network. Normally in different environmental pressure, the bacteria, they produce polysaccharides. And these polysaccharide is nothing but a carbohydrate. So we have different use of the polysaccharide, but other than that, this polysaccharide biopolymer help the bacteria to get adhered to a substrate. When the environment is extreme, the microorganism need to adhere to a substrate so that not to be washed away in the environment. After getting adhered to the substrate, what they do, they basically multiply, they divide, and they make the colony. And when they form this biofilm network, that gives the microorganism additional protection against different stress factors. Can be temperatures, salinity, ultraviolet ray, different toxic compounds, can be even antibiotics or osmotic stress. So when we normally go to collect samples, can be sediment, can be water sample from hot springs. Few factors that according to me are really important to take care of. We should use, while sampling, we can use some nets. Some nets can be of polyethylene, polycarbonate, and we can pin them in the sediment, and after some days if we go to take them out from the environment, they will have some microorganisms attached to that. So will be relatively easier to cultivate them from that kind of environment other than bringing them to lab. So we are giving them a similar kind of environment to grow. Another factor is homogenization. Often for homogenization, we prefer a saline solution or a phosphate buffer solution, physiological pH. So when we get the sample, and the sample is a sediment sample, it's often necessary to homogenize them after using some rotation for some 10 minutes, 30 minutes, depending upon the heaviness, depending upon the composition and the clump of the sediments. Another factor importance is the temperature. Often it's difficult to get the temperature that the microorganism go in that particular environment, but we can mimic that kind of temperature in the laboratory. And another factor is the incubation time, because often the microorganism takes very long days to grow. So initial days to determine the exact temperature and the incubation time is important, because often the fastidious microorganism grows in really longer time. So some normally what we use in laboratory, we supplement the nutrient broth, all the LB broth, Luria Bartani broth with specific media. But other than that, we can also choose some other media. It's in general, not even we don't consider hot springs. It's difficult to cultivate microorganisms that are in the extreme environment because of their nature. They are basically growing in a low nutrient condition. So different media that can be used for their growth can be R2A medium. R2A medium normally we use for quantification of bacteria in potable water also. But other than that, it is widely used in cultivation of thermophiles, because in general R2A medium is a low nutrient medium that gives a similar kind of nutrient medium to the microorganism. Another, if depending upon the salinity of the ecosystem, it can be fumarole, it can be hot spring. A very common nutrient medium is marine broth. Now, the difference between marine broth and R2A medium is marine broth have different salt supplemented with marine medium, and R2A medium normally we don't have the salt supplemented, but we use the sea water in that. So that's a little difference. Also, we use minimal media for defined growth. We can add different factors defining the growth of particular microorganism. That is different in case of different microorganisms too. Another is tryptic soya broth. it's a nutritious medium, obviously, but for growth of aerobes or facultative anaerobes, tryptic soy broth is an option. And in some cases, in case of the hot spring, sometimes the pH is acidic. So in that, or in case of fumaroles also, so in that case, yeast malt agar can be an option for the growth of acidic bacteria. And other than this, so when we are using all these different kind of mediums, we need to see in which medium it's growing. So it's like a probability. We have to try with various medium in what it's growing, and for getting the best growth, we can use the nets, we can homogenize the sample, and definitely we can modulate the temperature and the incubation time. So in one of the work that previously we have done, we have found a polysaccharide from one thermotolerant bacteria from hot spring of India. It was showing some antioxidant and emulsificant activity. So this hot spring that we have earlier talked, this was not before us. We have first reported some bioactive compounds from this hot spring. The temperature was nearly 66 degrees centigrade. That varies according to the different time period of the year. The pH is nearly 7, and the high arsenic was quantified. We have done some other works also related to the arsenic content and some arsenic resistant bacteria, their bioremediation. However, in this work, we have focused on this bacteria that was producing high amount of polysaccharides. It was nearly 8 gram per liter, 7.66 gram per liter of dry weight of polysaccharide was extracted when we supplemented the nutrient broth with glucose and peptone. So the substitution of the nutrient broth is important when we are trying to produce polysaccharide, because in different bioactive compounds, the supplementation is different, because in case of polysaccharide, the microorganism need a carbon rich medium. That carbon rich medium can be, we supplement the medium with carbon sources from outside, or another can be we use some residues, maybe some sugar rich residue from agricultural waste, can be sugar cane, can be beet sugar. So the agricultural residues that are rich in carbon source, there are reports that microorganisms are able to utilize that also. So that in a way basically decrease the production cost of the polysaccharides. So here the temperature was an important factor, and the arsenic. So what we found that before this, normally there are a lot of report on lactobacillus group, and this lactobacillus normally produce dextran. So in bacillus, before there was not much, there was report but diluted report. Some were reporting that there is glucan type of production, but specifically it's a levan or a dextran, because it can have alpha 1,6 bond, it can have beta bond. So depending upon the glycosidic linkage, it's dextran or levan was not before understood. So we perform GC, and we also get only glucose as the predominant monosaccharide. But then when we did the NMR from the, it was a low molecular weight polysaccharide, but with the NMR, we get the confirmation that this was basically a dextran type of polysaccharide, that we different peaks that we found between 3 to 6 ppm. And interestingly, this was also a thermostable polysaccharide. Normally the bioactive compounds that the thermophile produce additionally have the thermostability. So often we prefer them to use in environment, in industry, like for example in case of textile industry, often we use amylase, and this kind of textile industry enzymes often needs temperature stability. So this kind of polysaccharides give them the environment to embed them into the polysaccharides, so that they have a long time sustained release. In the food industry, often they have long-term use, because in food industry the additive that we are using are synthetic in origin, or often the additives used in food industry don't have the additional thermal stability. So the polysaccharide produced by the normal microorganism mesophiles have the thermostability near to 20 to 40, but in case of thermophiles, that's more than to 60 to 70 degrees centigrade. Till date, there is the highest report of 294 degree from a geobacillus species from a Bulgarian hot spring. Geobacillus, this is till now the maximum thermostability reported from any bacterial species. So we have tried to get its bioactivity, and we have seen that it was, when we added sodium chloride, when we make a saline polysaccharide solution, it was showing thermoresistant and shear thinning behavior. This kind of behavior are really important for any biomaterial to be used in food industry, because the texture of a food is important. For example, when we are taking a yogurt, we prefer a yogurt that have that sticky nature, not something like very watery. So greater hysteresis area, better structural reversibility, and more that phase transition temperature is, more better we can use that compound in the industry. Actually, long around from 1972, the work of Yellowstone National Park, thermophiles has been exploited or have been explored long for their different industrial usage. And the positive thing of using any biomaterials is they are biocompatible and they are bioreversible. So we are not having any secondary residues present in the environment when we are using them in an industrial procedure. Also, this polysaccharide had good emulsification activity against vegetative oils compared to the controls. So in general, we can say that the heat-loving bacterial polysaccharide are an inspiring alternative in food industry as antioxidant or emulsifying agent. We giving different example on where we can explore thermophiles. Going to give another example, the work that we have done, we have used a Pseudomonas species, and it was also producing polysaccharide. The thermal stability was more compared to the another one, was still 29 degrees centigrade thermal stability. The interesting thing is Pseudomonas is already well known for producing biofilm. They produce a really complicated network of biofilm. We have only focused into its polysaccharide, so we have purified the polysaccharide out of the biofilm. So when we focused into it, we saw that it's producing basically a rhamnoglucan type of polysaccharide. This kind of rhamnoglucan has been earlier reported in plants. Plant basically produce rhamnoglucan type of polysaccharide, but this was one of the report where bacteria, there are only a few report where bacteria produce rhamnoglucan. In this case, the Pseudomonas was producing rhamnoglucan. Actually, even if the species is same, often it has been seen that when we supplement the microorganism in their growth medium with different nutrient sources, they produce different kind of EPSs. There are papers related to this, that when we defer the residual agricultural residue for the growth of microorganism in different agricultural residues, it's producing different chemical nature of the polysaccharide. So even if it's a Pseudomonas or it's a Bacillus subtilis or it's a Bacillus anthracis or any bacteria, they produce different kind of polysaccharides, even being the same strain, depending upon the supplementation that we are giving or depending upon the genetic setup, the environmental makeup that they are having. So this normally how we decide that which polysaccharide can be used for food industry, which polysaccharide can be used as a biomaterial. When the polysaccharide network shows a plastic kind of field and more conjoined, they are not like agglomerated lumps separated from one another. And instead of that, they are more showing like a plasticized feature, like a material. That's the kind of polysaccharide that is expected to be used as biomaterial for even nanoparticle synthesis. Because when in case of polysaccharides, they have the free carboxylic group, free hydroxyl group, many negative charges, open negative charges, and the metal ions have the propensity to interact with them, a natural propensity. So often the polysaccharide are favored for green synthesis of nanoparticles because you don't need to use any reducing agent, you don't need to use a chemical reducer to form it, and it's giving a one-step synthesis procedure. In this case, we have used silver and we produce silver nanoparticle, but in general the polysaccharide was showing a plasticized film
It seems that we have problems with the connection of a partner. Let's see if she will try to connect again. We are trying to contact a partner to see what happened. So I don't know, Bo, if you are still there, maybe you can talk about your presentation a little bit more.
I'm here. I was just answering because there was one question in the chat about the Animox.
Okay, good.
The Animox depth and from Carlos Enriquez and asking whether the maximum high Animox activity that we see relatively shallow in the oxygen minimum zone, whether that could be related to the secondary chlorophyll maximum there, but not directly, because the at least it's offset, so that the peak in Animox activities is deeper than the secondary chlorophyll max typically. It's not, they don't coincide. Of course, there could be production in the secondary chlorophyll max, which then sinks and could then be driving some.
What point I got cut?
Okay, Aparna, you come back.
Am I was cut just a few minutes back, isn't it? Because I was talking.
Yes.
Yes. Okay. I'm going to present. I really don't know how it happens that suddenly the Wi-Fi got off, and I don't know.
Okay, maybe the connection was wrong. Okay.
I think I was here, isn't it?
Yes.
Okay, thank you. So the yes, here. So the microorganism was the polysaccharide was showing good antimicrobial activity against bacteria and both fungi. So it was showing in general a broad spectrum antimicrobial activity. So that's a proof that the bioactive compounds produced by the thermophiles can be used in general as an alternative of biocidal agent to in this time of multi-drug resistance. So we can have an example and in general conclusion that heat-loving bacterial polysaccharides are also inspiring alternative in the pharmaceutical industry as antimicrobial nanoparticles. Now coming to the work that we are doing in Chile, in the Fondesit initiation that I am working with. In this Fondesit initiation, we are focused on using the thermophilic bacteria that are in the Maule region and has not been explored earlier for bioactive compounds. We are using these thermophiles for using polysaccharides. So that's one of the few work that we have published till date. Initially, we have got a Bacillus hyanacea. Bacillus hyanacea has been earlier reported from marine environment, and Bacillus hyanacea in this case we got from this hot spring of Maule region, Campanario, and this Bacillus hyanacea was forming good amount of biofilm in increasing temperature till 60 degree. Actually, was producing best biofilm in 50 degree centigrade. So was and it was not hampered with carbon source. Actually, nitrogen source was making a big factor in the formation of biofilm. So it was a good result that we have got, that not only carbon source are always important, often nitrogen source is important for the formation of bacterial biofilm. So we use different mediums, nutrient broth, LB, marine broth, and marine broth it additionally supplemented with salt. And we have seen that the formation of biofilm is very strong in case of marine broth. The biofilm network is more thick and compact in structure in case of marine broth, as you can see also in the image. And then we have produced polysaccharide by these bacteria, and we optimize the production of the polysaccharide by statistical optimization. And when the predicted was nearly 6.05 gram per liter, it was showing around 5.98 gram per liter. So the statistical optimization was also fine. So that's how the polysaccharide was looking, produced by Bacillus ayanacea, was also showing a plasticized film-like feature, and the temperature stability was also high, as we can see from the thermal stability analysis. So we did a more in-detail study, because from the 1H NMR we have not been able to elucidate the total structure of the polysaccharide. So we see that from the chromatographic analysis, it was having mannose, glucose, and galactose. And finally, we have that idea that it was showing a production of, it was basically a mannosilated polysaccharide having some glucose and galactose. So it was showing 4:3:0.3 molar ratio of the polysaccharides, and having a glycosidic bond of alpha D mannose, 1,4-glycosidic linkage. So that we had the idea doing the 1H NMR and 13C NMR and 2D NMR analysis. So after getting the idea that it was basically a polysaccharide composed of mannose, glucose, and galactose, and not a defined polysaccharide like rhamnoglucanon dextran, it was a novel type of polysaccharide, we tried to see its antioxidant capacity. We have seen that it was showing good antioxidant capacity compared to the standard antioxidant, and also was showing activity as bioflocculant, and was showing emulsifying activity also against a broad range of vegetative oils. When you compared with the traditional biopolymer that we normally use in the food industry, xanthan gum, in often cases it was showing more activity than the xanthan gum. So all the activity that it was showing, bioflocculant capacity, antioxidant capacity, emulsification capacity, all these are the features of being used in food industry. And again, we proved the same fact like earlier, that the thermophilic polysaccharide have a potential use in the food industry. We got another bioactive compound from the same bacteria. When we changed the medium, we increased the salinity, and the microorganism was produced, the same bacteria was producing melanin. Melanin have been earlier reported by different bacteria, by Pseudomonas, by different bacteria. In this case, we were first showing melanin production by Bacillus ayanacea, and it was showing autofluorescence capacity. We did the entire structural analysis, and we confirmed that it is melanin. Confirming the structure of melanin is often difficult, because melanin have 5 different groups, pyomelanin, eumelanin. So it was really difficult to do, but we have proved that it is a melanin, and a pyomelanin type of melanin most probably. And again, melanin already is reported to have good antioxidant capacity, and our sample was also showing good antioxidant capacity compared to the ascorbic acid. Actually, the melanin was showing nearly 99% radical scavenging activity compared to the ascorbic acid, and was showing really good antioxidant capacity. So we can say that the heat-loving bacterial polysaccharides produce antioxidant bioactive compounds also. And in these days, the industry need antioxidants that are biological in origin and non-synthetic, so that we will not have a secondary residual impact in the environment. And finally, we have not started this work yet, but recently we have won a project together with Alexander Galan, my colleague, my another colleague Alexis Castillo and Alex Echeverria from UCM. We have national team composed of researcher Gustavo Cabrera from Concepcion, Paris Lavin from Antofagasta, and we have also have international team from Spain, from China, and from USA. And this time we are focusing on bioactive polysaccharide as food additive from Deception Island, Antarctica. We are funded with INACH, the Society of Antarctica in Chile, and we are trying to see, this time we are trying to see in a more complex way the polysaccharide production. So rightly we have Alexander Galan, who have that area of seeing the polysaccharide production from the point of view of biogeochemical cycles, because the biogeochemical cycles have an impact on the microorganisms on their polysaccharide production. So we are trying to understand the feedback and response of the environmental factors. Understanding the environmental biogeochemistry, we want to produce the polysaccharide, we want to characterize it. Also, we want to characterize the environment that the microorganism is. And after producing the polysaccharide, we want to, after structural characterization, complete structural characterization, we want to see its application as food additive. So that's something that we will do in the next time, because till now the Antarctic expedition have not started. So probably we will start it very soon in this December or January time. So thank you all, and I'm thankful for all the results that I have seen. The part of them are from my Fondesit initiation. And the result was mostly done, the melanin work was done by one of my thesis student of undergraduate, Catalina, and the rest work of the polysaccharide was done by Patricio and Ignacio. So really the young students with full of energy, they have done this work together with me. So I'm really thankful to them. And other than that, I'm also thankful to Fondekip that I'm co-investigator, and we have got a lot of beautiful confocal microscopy image. The Fomento in Vinculación for this biogeochemical cycle network in BioNet, and all the people who are associated with it from Iraq, from India, from Chile, everyone who are associated to it. Thank you so much. And also I'm thankful to my laboratory, Senbio, where I'm getting the laboratory support, and my center, SEAM, the biotechnology faculty that the students are in, and also the University of Bardwan for the part of my PhD work that I am showing. Thank you so much.
Thank you, Parna. So this is an interesting topic to explore, and especially doing this project in Antarctica. So maybe we have a question from Carlos here.
Hi, Aparna. Do you hear me?
Yes. Hi, Carlos.
Hi, I have a question regarding the melanin production that I think that's really relevant topic, and how it is possible to have like a big batch culture of a specific microorganism that can produce this melanin in a great scale to really produce this compound, or is necessary that the bacteria form like a consortia, and the biofilm is necessary absolutely to having large amount of the compound? It's how you can handle with these two possibilities.
Actually, in this case, the biofilm production and the melanin production was two separate factors. This bacteria, Bacillus ayanacea, under salinity stress was producing melanin, and under temperature stress was producing polysaccharide. So they both were not interrelated to each other. So when we increase the salinity, the microorganism was producing the melanin in a really high amount. It's not like polysaccharide that's some gram per liter. So I think I'm also looking into addition of phenylalanine, L-phenylalanine, because often inducing L-phenylalanine to the environment, to the culture, induce the production of the melanin. So till now we have not done that, but we have got a good amount of production of melanin. But that time when we got it, this was something really new, because earlier bacillus have not been reported to produce melanin, and that too for some stress, because when we are augmenting the salinity stress, so it was producing. So it was producing in good quantity, but in order for a industrial point of view, if we augment the production, probably supplementing the media with L-phenylalanine can be a good option. And I think for that we don't need a consortium, because this bacteria itself is producing the melanin. So the biofilm production or the consortium is not.
Excellent. Thanks, Aparna.
Thank you, Carlos.
I have here another question from my colleague Alexis Castillo from the Catholic University of Maule. He wants to know which criteria you consider to select the thermal pools that you pretend to sample in Antarctica.
Okay. In case of Antarctica, together that we have decided that we will look into, actually there is a paper of Sluice et al. That's back in 2000. That was one of the preliminary work in Deception Island fumaroles, and they have got more than 10 different points to work with. But in general, in this proposal that we are thinking to project on is minimum disturbances with human, because also Deception Island have the touristic disturbances during the summer period. So often we have seen that the production, when the environment is contaminated with human disturbances, the probability of getting the bioactive compound is less. So more undisturbed the environment is, we have better possibility of getting diverse kind of microorganism. So in this time we are focusing only on the environment that have less human disturbances, and probably if we are, for example, we are putting some polyethylene or polycarbonate film back down in the pool. So if someone is going to get sauna there, so of course it will be gone. So we have to focus on something that the temperature is high and also it is relatively undisturbed. So normally what my idea is that January is a really difficult time for Antarctica, because normally that tourist pressure is also there. So other than that, if we go before, there will be better possibility of getting more environment that is having less human disturbances.
Okay, and I have another question. This is not my topic of research, so I'm completely ignorant about that. But you need that the organisms, the bacteria that you work in, is stressed by the environment to produce these polysaccharides. That means it's bacterial that are not stressed doesn't produce that, or is not enough. And the other question is, how do you know what is the use of the polysaccharides that the bacteria produce previous to do the finding? I mean, you explore the bacterial fields, you found that they stress and they produce the polysaccharides, but how you determine what is the exactly use for that?
In case of bacteria, that what we are looking into for how we are getting the idea of where we will use. Okay, first about the environment. The environment is not, there can be two factors. There is some stress. The stress can be temperature, can be salinity, can be antibiotics. Because imagine, for example, we are now in Chile, we have cold temperature a little, and we prefer to be in the home wearing a jacket. But in the summer, so we prefer to be under a protecting cap. So in case of bacteria, it's kind of same. They want to be inside a colony that is protected with some polymer. So it's giving them an extra protection against any extreme factors that they are having. And in that factor, they are forming a biofilm. So no stress factors are able to enter inside their colony. That's one thing. So either when we produce the bioactive compounds using the bacteria can be polysaccharides. So we normally try to mimic the same environment that was there in the normal environment. For example, if there was high temperature, we try to mimic the similar kind of temperature in the incubator with similar kind of medium, with additional supplemented salt or something. Of course, it's never possible to mimic the exact same medium, but as similar as we can do, so that they feel that they are in the similar environment. And there is a factor that often for this, after a lot of cultivation, maybe after 100 or 200 phases of cultivation, bacteria lose the capacity of producing bioactive compounds, because that time they are not exposed that much to the environment. So slowly they lose that capacity. And the second factor about how we get to know the idea of where we can use it, normally the polysaccharides are either in the microscopic feature, either they show a plasticized film-like feature like a biomaterial totally, or normally they show diffused macromolecular lumps. So when these are diffused macromolecular lumps, normally we want to use this kind of polysaccharides for food industry or for wastewater treatment purposes, because they have the flocculation capacity and they flocculate the entire residues to precipitate out. But in case of, for example, when we are trying to use the biomaterial as emulsifying agent in the food industry particularly, so it need to have a texture, and that texture only comes when it have that plasticized film-like feature. Or for example, in case of nanoparticle production, the nanoparticle need to be embedded in the materials, so it's giving the additional grafting capacity. So before, we cannot have the idea when we do a lot of sampling and when we have a lot of different polysaccharides, needs, then we can focus a few of them for our application. But seeing the environment, we cannot have the idea that what application it can have.
Okay, thank you, Dharma. We are on time in this moment, and I hope that you have successful in the Antarctic sampling, and I hope to...
Together we will. We are going all together.
Okay, we have now 15 minutes for a coffee break. So we come back at 11:30 with the talk of Dr. Sara Cuadros. So see you soon. Okay, welcome back. Our next speaker is Dr. Sara Cuadros Orellana. Dr. Cuadros is a full professor in genomics and informatics at the Catholic University of Maule. And she is a member of the directories of two national scientific societies, the Chilean Society of Microbiology, and the Chilean Society of Bioinformatics. As a researcher, she has been studying the microbiology of extreme environments, such as saltwater hot springs and acid mine drainage in the past 18 years, focusing mainly in understanding ecological evolutionary aspects of extremophilic communities together with their biotechnological potential. Dr. Cuadros has developed novel biotechnology solutions with medical and food industry applications, and among her main achievements are a patented device for bacterial meningitis diagnosis and antimicrobial hydrogel that considerably extends the post-harvest life of fresh fruit and vegetables. She's currently the principal investigator of 3 national grants, namely a regional FIG, a funded regular, and a funded kit median, using cutting-edge DNA sequencing and cell cytometry methods, some of which she will present to us today. So, it is a pleasure to introduce Dr. Cuadros. So, Sarah, please, the public is now yours.
Hi, good morning, Alex. Good morning, everyone. Thank you for inviting me to this workshop. It's a pleasure to be here. Let me share my screen and present you some of the research. Can you see my presentation?
Yes.
Okay, great.
Sarah, put in the presentation mode. Okay, now.
Okay, so my name is Sara Cuadros. I'm a professor at Escuela de Ingeniería en Biotecnología de la Universidad Católica del Maule. I'm presenting today my research on microbial ecology, and I'd like to emphasize the importance of single-cell sequencing to the improving of our knowledge in this area. So this is what my talk is about today. So just as a brief introduction, I'd like to mention that microbial communities, they are usually quite complex. They are multi-domain. There's a lot of intra and inter-specific variation among the genomes. There's a lot of taxonomic diversity, but also functional diversity, but also in some environments, there's a lot of redundancy. So sometimes different taxa can occupy the niche, the ecological niche, and we see our functional redundancy regardless of the taxonomy assemblage. So also there's a highly dynamic interplay among the species. So every sample that we take to study the microbial community is just a print screen of the reality. It's quite dynamic, and so if we do not do a temporal follow-up of the environment and take samples at different times, we will not see actually this dynamic behavior. So in here, we can use different approaches. So I will mention some of them that we currently use and that we intend to use more extensively. So this picture depicts what we know about the microbial diversity nowadays. You probably saw this iceberg picture several times, indicating that we just scratch the surface of what is there in the environment, the diversity of microorganisms that exists. And we are now very aware of the amount of data that we can use to study this hidden diversity, which is also often called the microbial dark matter. which is the main part of this iceberg. So we realized that, and we try to use different methods to reach this huge amount of information that will probably allow us to progress and understand better the microbial systems. So I'd like to mention that rare taxa, which are those that occur in very low abundances, so they are the main part of the big part of the iceberg. So lots of different rare taxa, they are here in this portion that we seldom reach through some, depending on the approach that we use. So they are not so accessible, difficult to cultivate, but they are probably most of these microbial dark matter. Also, sometimes they can have a very important role in the ecosystem. So they sometimes are the major together, all these rare taxa can be major drivers of the multifunctionality of an ecosystem. So they equilibrate this scale because of their important functional role. Also in some biological processes, some specific ones, they can play an overproportional role, which is also relevant when we are looking for biotechnological applications. So I put here some references that support these comments. So also microdiversity, which is what I mentioned, that the genomes vary intra-specifically a lot. This brings us to the concept of pangenomics, which is has been discussed in depth, and we have now recognized this importance in microbial communities, and also made us rethink the idea of a reference genome. So any strain that we isolate and we call it a type strain, and we deposit it in a culture collection, is of course a reference strain. Its genome can be a reference genome, but what's the meaning of that, considering that we have so much variability among the genomes of a same species? So we need to understand that each strain represents a single individual. Sometimes we can consider the pangenome as a population consensus, so the sum up of all the genomes of a similar species will represent its pangenome. Then we can talk about the functional importance of this pangenome, and we also can think of minimal and maximum genomes. So we can understand then what's really core, central to all the genomes, so the core genome, and also what's more related to adaptation to very specific micro niches that each strain can colonize. So this microdiversity is important to understanding not only the evolution, how organisms adapt and evolve, but also how they adapt to changing environmental conditions, which is very important to what we are talking today in this context of global warming and environmental change. So this is something we are very interested in studying more in depth. So choosing the right method for studying the microbial communities is very relevant to what we can conclude regarding this microdiversity. Some methods are very good to understand the composition, the taxonomic structure of a community. Some other methods allow us to reach and understand the functional aspects. So what is happening in this ecosystem, what biogeochemical cycles are taking place or can take place, because we see the genes and the potential, right? And some other methods will allow us to understand the microdiversity. So this is what we are currently interested in. So regarding what we know about microbial diversity, I can show you here one proposal of a new tree of life in which we see the archaea here. The archaea is this small branch here to the bottom together with the eukaryotes. And here in this tree, actually bacteria are highlighted as a main domain, likely divided into two, actually. So basically we can see 4 different branches, right? And these calls, this is actually what I was explaining, the biggest part of the iceberg. Each red dot represents groups or branches of the tree that has no cultivated isolate. So they are only known through the metagenomes. So what we sequence from the environment, we know they are there, but we can't cultivate them. So we can't characterize them physiologically. We can't understand most of its genome, because part of the genomes that we recover code for hypothetical proteins. We know that around 20 to 30% of a genome codes for some proteins that we don't know what they do. So we really need to go further, to push further into the methodological aspects of microbial ecology, so to get access to this information and finally generate better databases and be able to understand more in depth all these communities, all this diversity. Okay, so for instance, this big group that has plenty of red dots, this is CPR, the candidate phyla radiation. Most likely these organisms represent symbionts of other bacteria because they have small genomes, they have limited ability to synthesize basic molecules, so they can't be isolated except if we can provide them all the necessary requirements for growth. And this is quite challenging, actually. So designing good enrichment media and methods for cultivation is quite challenging. So understanding their genomes can guide us on isolating them in the future. So cultivation-based methods and cultivation-independent methods, they are the 2 big groups of methods that we can use in microbial ecology. There are many more methods than the ones I listed here, but this is just a general idea of the main ones. So when we talk about cultivation-based methods, we think about strain isolation. So when we get a sample, we bring it to the lab and we incubate it and we try to recover a colony. We can also do this in a directed way, targeted way, enriching the sample in a medium that we'll select for some characteristic that we want. Currently, people are doing high throughput isolation, so trying to get the most diversity possible. So the high throughput methods, they are usually miniaturized, so in droplets. Cells are isolated and cultivated so to form microcolonies, and these are then brought into culture using different types of approaches. So also the low and high throughput screening of these new strains, right? You can characterize one by one, or you can do this in a format that allows to analyze thousands of cells at a time. Well, on the other hand, we have the cultivation-independent methods. These are the classical ones that use indirect measurements, biochemistry, directly in the environmental site, such as respirometry and other in situ techniques. Also, we have molecular biology methods that are classically used, like DGE, RFLP, and other fingerprinting techniques. But mainly, nowadays, microbial ecologists are basing on their research on sequencing, so high throughput sequencing. This brings us to this group of methods that include amplicon sequencing, which is also called metataxonomy, metagenome shotgun sequencing, which is the one that I use the most in my lab, and single cell sequencing, which is the one I would like to introduce here as a new tool for microbial ecology. Actually, not so new, but it's getting more and more important. So why are cultivation-based methods limited? Because I mentioned before, the complex nature of ecological relations like symbiosis and all others, and also the intrinsic limitations in cultivation methods because of the conditions that we can use, usually prevents the isolation of relevant organisms. Well, so the another information is that an estimated 99% of bacteria and archaea are unculturable. So this has been repeated over time and based on some observations that most of the organisms that we see through metagenomics, they will not be brought into culture easily. So the low throughput methods, they can't reach the in-depth, this iceberg picture. They just scratch the surface. The high throughput, they go a little further, but still are limited. So the cultivation independent methods, they have some, they have been used widely. They have also some methodological requirements for being useful. So the first one is sampling method. And here in this workshop, we propose to discuss this sampling method and design. So getting the sample in a reproducible way, preserving the samples correctly to get to the laboratory and really get the good picture, a good reproduction of what the community is and not letting it change over time is very important. Also the metadata recording, so checking all the environmental parameters inside and later on. So preserving the sample to perform chemical analysis, everything is important here to get a clear and accurate picture of the microbial communities. So this is to start with. So cultivation independent methods, they are very powerful, but they are very sensitive to this type of aspects. So sampling is important and metadata is very important for drawing conclusions as well. Well, all cultivation independent methods based on sequencing, they depend on getting a complex sample, extracting the metagenomic DNA, then generating data and analyzing it. Most methods include NGS sequencing and bioinformatics. Some of the data that we can generate are amplicon sequencing, which is the most used method nowadays to get access to the structure of a microbial community. The principle, the general principle of amplicon sequencing is that you design primers that will tag or target conserved regions. Then the amplicons will cover variable or hypervariable regions, which are very informative. You can place the organism in a phylogenetic tree, for instance, and then you can define the structure of this community. To some extent, you can also draw conclusions and infer functional aspects of the microbial community. For instance, if you find an organism that is photosynthetic, you can infer that this is a function that is being performed by this organism in a community. So different marker genes are used for bacteria and archaea, usually regions of the 16S gene, and for fungi, regions of the interspacer region between the ribosomal genes. So this is depending on each focus of a researcher, right? If you are interested in prokaryotes or eukaryotes. Usually this is very specific of each study. So the deep amplicon sequencing is a very powerful method. It's cost effective because it's quite cheap to do it for many samples. And it's more sensitive than most methods, including metagenomics, the shotgun metagenomics, because you can get to the rare biosphere, these very deep into the low abundance taxa, because as it's based on amplification of a signal, it can really detect some taxa that are present in very low abundance. So this is a pro of this method, or these are pros, right? The cost effectiveness as well. But some cons are the lack of functional information, of accurate and complete functional information that's not inferred but directly seen, right? It can't really get to the complete scenario of functional diversity of a community. Also, there's an amplification bias in this type of methods, which is easy to understand since not a single primer can really target all the organisms. It's the best we can design, but it's not really universal. Other types of biases that are common to any type of culture-independent method based on DNA sequencing are lysis, so we can sometimes not completely lyse all cell types, and also that some genomes contain several copies of some marker gene. So this will cause an overrepresentation, which is not real, of the abundance of these cells. These are things to be considered. Well, shotgun sequence, on the other hand, can get deep into the understanding of the profile, functional profile of a community, and usually it's as sensitive as the sequencing depth. So the deepest you sequence, the more you learn about the functional profiles. In summary, amplicon sequencing will tell us who are there and what they likely do, and shotgun metagenomics will tell us what they do and probably most of the organisms that are there. So recently I've been to Lausanne to attend to ISME, the International Society of Microbial Ecology Symposium, and I brought this information today about the amount of posters and presentations related to different aspects of microbial ecology regarding the methods. So what I saw there is that amplicon sequencing is still being used by most researchers. As I mentioned, it's the most accessible method. Shotgun is still important, and the reconstruction of genomes based on shotgun data is also increasing. We saw many different presentations, but single cell was definitely one of the most important methods discussed there. 2 companies were presenting new solutions for single cell sequencing. 10 sessions were dedicated to presenting different aspects of microbial ecology, and single cell was used as method in all of these. And 91 posters, so it's a really big amount. So I was really happy to see that most people are trying to get into single cell sequencing, developing new methods, very creative ones, and we will try to incorporate these to our research as well. So briefly, I'd like to present you some examples of use of single cell and how it improved our understanding of microbial ecology. So one example, I presented it before, I will try to be brief, is just a very complete project intended to understand microbial communities in a mining environment. So this is a copper mine in Brazil in the middle of the Amazon, and the company asked or hired us to study these environments and to try to understand microorganisms, which microorganisms are related to acid mine drainage formation, to bioremediation of these acid mine sites, and find this biotechnological potential in the same environment where they exist. So we did a site recognition expedition, and we measured many types of different compartments in the mining area, and we designed a research based on that, and we could collect samples in different types of environments, mainly mine tailings, which is a dam where they throw the rest of the ores that they cannot anymore extract. So they are trying to extract copper. and what's left is thrown to this tailings dam, and the acid mine drainage, which is a problem that happens spontaneously in some areas. So we try to design an experiment to understand what happens between seasons, so dry season, rainy season, which could affect the ecology in the mining tailings. And in the acid mine drainage, we didn't compare different seasons, but we did a complete analysis of surface and sediment samples. We collected different membranes with pre-micro 0.80, 0.2. And in the tailings dam, we collected water from the surface, from deep water, which was in this case 15 meters, and sediment, which was 20 meters deep. So we did tool benchmarking for bioinformatics. I will not go into detail here, but it's very important to test your methods, bioinformatic methods, before you decide on which to use. We did this full pipeline of shotgun metagenomics, amplicon sequencing, reconstruction of MAGs, and also single cell genomes. To compare the methods and get them all the results integrated into one big database to be able to understand completely this type of environment. So comparing amplicon sequencing and shotgun, we could find, of course, that we can get into taxonomic information. We can understand, for instance, that sediment samples have much higher diversity of species than the water samples. We could understand that higher carbon and nitrogen, which happen in rainy seasons, could increase some types of taxa in the communities. So this information we can get using these methods, and so on, and which organisms occur in acid mine drainage in different sites. But what we saw was something interesting, that not a single method, not shotgun metagenomic, not amplicon sequencing, could see the full picture, because only 16 phyla were detected by both. 30 phyla, as shown here in this Venn diagram, could only be seen and detected by using amplicon sequencing, which I mentioned that since it amplifies the signal, it can see further. But 13 taxa were not seen by amplicon sequencing, but they were detected by shotgun sequencing. So this shows us that not a single approach is enough. We can often combine them to get the best scenario. And we did the in silico, like we tried to check if our primers really couldn't see all these 30 phyla. And we did this in silico check and compared to this database that contained at least one representative of each taxa. And indeed, it's very low. Some were not detected in silico. Some were very poor in very low sensitivity in detecting these taxa in silico. So confirming that really that we can't base on a primer to get all the taxa that are available. Some will not be seen by amplicon sequencing at all. So combining both approaches is a good option. So we could understand that some patterns occur. So acid mine drainage, different sites of acid mine drainage, these communities, they are more like each other compared to the tailings dam. And also the tailings dam has, of course, different communities in the water column and in the sediment. So all these communities can be separated using the principal component analysis. We can also compare the metadata to the information of the microbial diversity in each sample and understand which factors contribute to the differences that we observe. So in the water, for instance, of the tailings dam, we could see that calcium, copper, it would actually not only the tailings dam. Tailings dam are the first 4, and acid mine drainage is the 5th. So the blue graph represents the acid mine drainage. So copper is being solubilized in the acid mine drainage. So from copper sulfide to copper sulfate. And we can see this clearly. So this is impacting the type of organism that we can see. Also, some bioremediation was being used by the company at the time. They were using limestone to try to correct the pH of the acid mine drainage. So we could also see the effect of this remediation on the microbial composition of the site. We could assign specific functions in the ecosystem to specific taxa. So we can see which are the main players in each type of process. This is all possible through shotgun metagenomics. And also, we tried then to use these new approaches. At the time, it was quite new to use genome resolved metagenomics, which is the reconstruction of the MAGs, and the single cell genomics. So at the time, we did both. I will start by presenting some results on single amplified genomes. We isolated the cells through FACS. FACS is a method for staining the cells and sorting them according to the fluorescence. We can then lyse the cells, amplify their genome by MDA. MDA is a method based on hexamers. So they are not specific primers, and they will amplify the genome simultaneously in different points, and it's branching. So all the genome is being represented. And then you can bring 1 picogram of DNA from one cell to 1 microgram. So it's a really good amplification of the full genome, so we can have enough DNA for sequencing. This is what we did. We managed to recover some MAGs, the first here, except the later 2. These are all MAGs. And we were surprised to see that most MAGs, they were assigned to Chitinophagaceae, which is a group that we never thought would be found in an acid mine drainage. So we compared these genomes, we did a phylogenetic tree. They seem to form 2 clusters, likely 2 different species. But all groups, they also were similar to some extent to Hydrothalia flava. Well, Hydrothalia flava was available in a culture collection, but it had no genome sequenced at the time. So it existed at a pure culture, but its genome was not sequenced yet. So what we did was to sequence. We got the strain, we got its genome, sequenced it, and we used this as a reference to understand the SAGs that we had isolated and sequenced from our acid mine drainage. And this allowed us to see several important aspects. For instance, that these SAGs, which are similar to Hydrothalia flava, they are very specific to acid mine drainage environments. So here you see that the first 3 are tailings dam samples, and the later 3 are acid mine drainage environments. So the abundance of these SAGs, when we map them to the metagenomic reads of these environments, or we map the metagenomic reads to the SAGs, that we can see that they are clearly specific to acid mine drainage also here. This is acid mine drainage, this is tailings dam. So they don't occur elsewhere. Well, we analyzed these SAGs more in detail. We could understand aspects related to the microevolution. We studied the response to viral infection in each of these SAGs because we wanted to see how different they were, if they were clonal or if they were occupying different niches. And we could see some differences that point to they reinforce the groupings into 2 clusters, but they also bring information about the viral assemblage in this environment. For instance, here we see the detection of a CRISPR-Cas cluster in this MAG, also a different MAG, and here is a metagenomic contig. So we can understand. We could recover a metagenomic contig that also belonged to one of these genomes. and we could find synteny and all the same spacers that represent a memory of viral infection. So we can use either MAGs or SAGs retrieved from a microbial community as tools to understand microdiversity, and the CRISPR-Cas repertoire is a type of evidence for microevolution. So this is interesting, and it can be one of the aspects to be studied using single-cell sequencing. So we published this in molecular ecology. We saw that these Hydrothalia-like organisms, they have a possible role in the nitrification. They have hallmarks of metal resistance and horizontal gene transfer. And also, as I mentioned, the CRISPR-Cas repertoire that can point to an intense phage predation in this type of environment, which is very little studied in acid mine drainage. So the viral assemblages in acid mine drainage, they are not well known. So we can get closer to understanding this as well. Also the metagenome assembled genomes. So we could recover 244 MAGs from all these metagenomes that we sequenced over the 4 years of this project. And some of them were interesting because they were related to these SAGs. So the SAGs were related to Hydrothalia, as you remember, probably. And the two MAGs, we recovered two MAGs that belong to Patecy bacteria. This is a CPR. Remember the branch of bacteria that had plenty of red dots in the tree of life that I shown. These are probably symbionts, right? As I mentioned, because they have hallmarks of reduced genome size. And we recovered two MAGs that belong to those groups. And indeed, if we analyze the co-occurrence profile of these groups, they are also related to Hydrothalia, suggesting that they are symbionts of Hydrothalia. So we could recover through single-cell sequencing one organism that is exclusively occurring in acid mine drainage. And from the metagenomes, we could reconstruct two genomes that are likely their symbionts. So this is an example of how different methods can coincide and show the full picture of what's happening in one environment. So these two SAGs were quite complete, almost 99 or 92% complete. So these are very good contribution to the knowledge of this microbial dark matter as well. So the impact of single-cell genomics in microbial ecology is well recognized. So these are just two papers that highlight that. For instance, single-cell genomics of sorted nanoarchaeota. So you can target cells to sort and then analyze their genomes. Also here, the single-cell amplified genomes of two uncultivated members of the Delta proteobacterial clade. So this is a proof that we can go deeper and, of course, cell by cell, but we can also do this in a high throughput manner and get more information on groups of microorganisms that we can decide to study. So even more recently, Tanya Woik from the JGI in the United States, she published an article in which she understood more in depth the communities of hot spring using single-cell genomic. So it's very interesting because she also coincides with the view that different information that you can get from single cell, and you can't because she also did the metagenome and metagenome assembled the genome. So she recovered MAGs from the metagenomes, and she also did single cell sorting and sequencing. So she saw, for instance, a large array of mobile genetic elements in the single cells, which she couldn't recover from the metagenomes, probably because most of these mobile genetic elements, they have repetitive regions that can't be assembled. So when you talk about MAGs, you need the assembly step, and this is limited when it comes to repetitive regions. So one way to push further in this direction is combining short read and long read sequencing. And we are also trying to include that in our methods. So just to finish briefly, some of the projects we have ongoing. So I collaborate with some colleagues in the north of Chile, in Universidad de Antofagasta. We have studied this system, Lirima. So we collected samples from 3 ponds with different temperatures, and we did all the same approaches that I showed here until now to understand this environment. So I will go quickly through the methods, but I will show you the main highlights or the main results we got. We could make a complete metabolic reconstruction of the 3 ponds. So we know which process is present and or absent in each one. For instance, we can see the reverse Krebs cycle just in the hottest pond. We can also, of course, find photosynthesis only in the coldest ones, because the limit for photosynthesis is 72 degrees. So things like that make us understand better the limits of these processes, or at least the temperature limits for each process. We could analyze the cellular organisms according to their taxonomy. In my project, I'm really interested in archaea, so my interest is in the hottest temperatures. As archaea shown here in orange, they are very low, have a very low abundance in temperatures below 70. But higher than 70, they will increase considerably. And the very hot environments are the ones that are more interesting for me. So we can see the proportion, we can see the TAC archaea, for instance, increase according to the temperature and other patterns. Euryarchaeota will decrease accordingly as well. So we also found through shotgun data, not amplicon sequencing, some candidate clades of archaea. This is very interesting because by seeing them from shotgun data, we have some hope to reconstruct their genomes if we do more deep sequencing of the same sites. This sequencing we've done here was quite shallow, less than 1 gigabase per site, but we are currently working with at least 16 gigabases of each metagenomic sample. So we will definitely manage to recover more MAGs. In spite of that, we recovered 28 MAGs, 13 of which were of very high quality. And I just will cite some of them. So here I present the different ponds. This is temperature 42 degrees, 53 degrees, and 72. And there was some redundancy because these 2 ponds are connected. Even though they have different temperatures, they are just a shallow and a deep part of a same pond. This is not connected to the other 2. Okay, so redundancy is mainly present in these 2. So we recovered genome of Allochromatium, which is a photosynthetic sulfur oxidizing bacteria, actually 2 MAGs. 2 MAGs of Tepidimonus, which is a chemoheterotrophic bacterium. 3 MAGs of Thermodesulfitimonas, which is a sulfite reducing bacteria that has just recently been isolated and described. So we can contribute 3 more genomes or partial genomes to understanding this group. 3 MAGs belonging to Thermus, which is a caudoactive bacteria. 2 MAGs belonging to archaea, which have very few genome sequences available as well. And 1 MAG belonging to Ammonifex, which is a thermophilic chemoautotroph, able to grow by nitrate ammonification. Well, currently, we have focused in one project that I a grant that I got in 2020. So it's now the second year of my project. We are studying the same type of environment as Lirima, but in the Maule region. Here I show you a map of the Talca region with several volcanoes and active ones, where we find different types of hot springs with conditions ranging from pH 2 to 8, actually 1 to 8, and temperatures from 30 to 94. We are interested in the metagenomes and viromes, in characterizing the archaea, horizontal gene transfer, and archaeovirus host interactions. These are the main questions that I have. And we've been, and now the fun part that regarding the sites that we have been sampling. So some expeditions that we've done, I would like to show you some pictures. This is the Cordillera del Maule, and we have been there for 14 days above 2,000 meters above sea level, finding new hot springs and getting samples. These are some images, and I'd like to show you how they look like. They are quite hot, they are boiling temperature, and these are very extreme and poly-extreme environments, over 90 degrees, very acidic, and these are some of the environments we are doing metagenomics. Next, we will try to collect samples intended for single-cell sequencing, as I will mention in a while. These are more moderate temperature, like above 60 to 75 degrees, both also in the Maule region. Very big sites with a gradient of temperature, very interesting as a laboratory to study. This is the easiest site we can get. It's called Campanario. It's near Talca. We can go for the day. I took some students to learn how to get samples. And this is 54 degrees. It's not so hot. So not many archaea are present here. And this is one of the most extreme sites we visited. This is in the Cordillera del Maule. All sites are over 90 degrees. They are located over 3,200 meters above sea level. We found samples of 110 degrees at pH 1. And this is quite extreme. Actually, there's a volcano that is quite active. So there's a safety area in which we can't get near. So it was one of the most extreme sites I've ever visited. And now the Cordillera de O'Higgins, which is also covered by my project, we went to the Tinguiririca volcano, and we found so many solfataras, and there's more to visit. So we will go next there. So my last 3 slides, our strategy in these sites is to do the deep sequencing of metagenomes, thinking about the reconstruction of good mags. Also the hybrid sequencing, so using short and long reads to get better assemblies, and targeted and untargeted single cell sequencing. So for that, we applied for a Fondequip Mediano project to get more possibilities of developing our own protocols for single cell sequencing. So there's really big data under analysis. Our single cell sequence platform is now being installed. The equipment is already in Talca. We got a fax melody. It's a very nice equipment to work with. It's very easy to use. I visited some labs in Europe that use them, and they shared their knowledge with me, and they are quite eager to collaborate as well. So I'm really happy and hopeful that we will get very good data. Also, we got a Illumina MySeq, which is quite appropriate for single cells. The throughput it generates is quite okay for a single cell genome. And we already have the nanopore MinION that is up and running and has been helping us close entire genomes, because Illumina, of course, is very good, but it sometimes is limited in closing the genomes. So with that, I would like to thank my collaborators, the grants that have allowed us to do all this, my collaborators from Católica del Norte, Fundación Biociencia. These two are co-investigators in my Fundecit project. My colleagues from Argentina, Sofia Urbieta and Camila Castro, who are helping me do the fish packs of Archaea. Also the colleagues that helped me in the single cell aspect of my project. The two colleagues from Sweden, Claudia Bergin and Stefan Bertilsson, they received me in their labs. They also shared their knowledge. And Mark Musselman from the University of Vienna in Austria. So I'm really thankful to all these people who have helped me on the way of establishing new protocols and methods in my lab. So our team is mainly composed by undergrad students. I thank all of them for their contribution. Some are from the lab in Argentina, but they are probably visiting us. There's a PhD student from the mathematic modeling PhD program, who is also joining us in improving the binning methods, and also the support I get from Claudio Quevedo and Mabel Vasquez. So I thank you all for your attention, and I could take any questions.
Okay, Sarah, thank you for your great presentation. So there is a couple of questions in the chat. Maybe you can go there and answer because we are on the time. So I want to present the next speaker. This is Dr. Carlos Enriquez, a senior researcher at the Laboratory of Physiology and Marine Genetics, FIGEMA, of the Center for Advanced Studies in the Arid Zone, CEASA. Carlos is a PhD in biological science, mention of molecular genetics and microbiology. His research focuses on the study of the genetic and functional diversity of marine microorganisms in coastal environments. The main methodologies used are flow cytometry and massive nucleic acid sequencing that are part of the topics that he will present today. So Carlos, please, the presentation is yours. Thank you. Carlos.
Okay, can you hear me now? It's okay?
Yeah, it's okay.
Well, good afternoon, everyone, and thanks to Sarah for her very good introduction to microbial diversity. So it will help me to spend less time in explaining all the methodologies. So today I'm going to talk about the omics approach for the study of the biogeochemistry, specifically from the oxygen minimum zones. I'm not going very deep in the oxygen minimum zones because it has been already explained by Alex and by Bo. This is the typical graph that everyone you show in any slides. And for us, it's a model for the study of microbial metabolisms in extreme environments. As I mentioned, I'm not going to go too deep into the OMCs, as you've seen in previous presentations, but what is something that I want to let you clear that the minimum zones are expanding, and it is mainly as a result of climate change. So in terms of the oxygen, due to its positive redox potential, the molecular oxygen is one of the most important reactants in global biogeochemical cycles. So in this context, ocean oxygen loss can reshape the composition and function of marine ecosystems. So motivated in part by the fact that climate change is depleting the ocean oxygen, the recent studies are informing a deeper understanding of OMC's microbiology. So this research is facilitated by the advances in omics and sensor technologies. And these techniques are clarifying the processes and players that shape the OMC's biogeochemistry. So this is the same slide that Alex shows before. And we can observe that under oxic conditions, that is the more red colors, approximately more than 90 micromoles of oxygen per kilogram of water. Most of the energy generated via oceanic primary production is transferred to mobile predators. But as oxygen levels decline, as shown in the graph in the right, aerobic microorganisms escape to more oxygenated refuges, resulting in an habitat compression and a concomitant diversion of energy into microbial metabolisms. So typically the energy flows according to a well-defined sequence of reduction-oxidation reactions that was explained previously by Bo, and the order of which is determined by the amount of free energy available through each reaction. So oxygen, in this case, is the most favorably electron acceptor because it can provide more energy via its reduction than any other electron acceptors. On the other hand, CO2, the electron acceptor used in example by methanogenic archaea, yields the less energy. For example, in this case, as shown in this equation, the oxidation of NADH, the redox currency is more than 10 times more favorable in the presence of oxygen than CO2. So also this diversity of reactions that also can occur in aggregates like the particles as Marika knows. So the oxygen minimum zones also can present diverse metabolisms both in free living microbes and attached to particles. The secondary chlorophyll peak within OMCs is an ideal environment for studying microbial metabolism, and this is the site where my research has been focused as last years. And this panel shows profiles of temperature, the first one, oxygen in the middle one, and the chlorophyll, the last one, obtained from marine gliders that are like torpedoes that give us an idea about the distribution or dimensional distribution of different parameters. And it's also a heat map colors in which lower colder colors like the blues one represent lower temperatures in the first panel, and red color represent high temperatures, and the dotted line represent the mixed layers, and you can observe that it's really a gradient in temperature, but this is more evident in the oxygen, where you can observe a really low level of oxygen around 100 meter depths. And this oxygen minimum zones have really particular characteristics, like the high concentration of chlorophyll that is observed in the lower panel in subsurface waters that is inside the low oxygen waters. This concentration of chlorophyll can equal the chlorophyll of the surface that is around the mixed layers. So my research focuses on this chlorophyll maximum that is below the mixed layer and within the OMCs. And this is something that we are working on this time, and you can see like diversity indexes, and the diversity of the different microorganisms, in this case is bacteria in the upper panel, archaea in the middle, and eukarya in the lower panel based on barcode sequencing. We can observe that the diversity varies along the oxygen gradient. For archaea, this is quite clear that the great diversity is observed in really anoxic waters, and the diversity in terms of friction and Shannon index tend to decrease as we are going to surface oxygenated waters. This pattern is opposite to the pattern observed for the eukaryotic counterpart, that is the panel G in this case, or the lower panel, when you can clearly observe that the diversity of eukaryotes or small eukaryotes tend to increase when we are going from anoxic waters to really oxygenated waters. So this is very interesting that there are kingdom differences in terms of diversity that is related to the oxygen concentration and also the capacities or metabolic capacity that any kind of organisms have. But interestingly, when we are observing the diversity of the bacteria, we can clearly demonstrate in this work that the diversity of bacteria is quite different than this gradient observed for archaean bacteria, and much of the most richness and is observed in the deep chlorophyll maximum. Why? This is what we are trying to decipher, because this probably the deep chlorophyll maximums correspond an intermediate state and where can coexist aerobic and anaerobic metabolism. This sounds a little bit contradictory, doesn't? Because why oxygenic photosynthesis occurs where there is no light, or oxygen production in an environment that is supposed that doesn't have oxygen, or oxygenic photosynthesis in much deeper depth than the mixed layer. So this is a really very interesting system in which we have been working, addressing the biogeochemistry by different perspectives. And so what approach do we use in the laboratory to study the OMC's biogeochemistry? We have to keep in mind that for the study of microorganisms and their influence on biogeochemistry, as we are moving away from oxygenated environments, we must forget about the possibility of having cultured organisms. So all techniques, as Sarah mentioning, must be based on the genetic material. So in this context, when we're working in the environment, we can have some community questions, no? And in this context, so we can study which microorganisms are present in the community that is targeted or metabarcoding analysis through universal markers such as the code for the 16S RNA, ribosomal RNA, or 18S or ITS, or even some functional genes that can serve as taxonomic markers. Or we can study what is the genomic potential of the community. As Sarah mentioned, I'm not going further on that. And in this case, we can use metagenomics or whole genome sequencing. And but also we can study which of these microorganisms are active in a particular environment. And this is what we call it metatranscriptomics. And we can study the messengers in the case of generally we study messengers for eukaryotes, or for bacteria that is quite more complicated because the amount of transcript that we recover when do metatranscriptomics is really low, but it's also possible. So we need to do some ribosomal depletion to have a sufficient amount of transcripts, and we can observe the activity of microorganisms. So we can combine these both approaches to have an idea about the metabolism of the community, the phylogeny, if there exists lateral gene transfers, and all the possibilities that Sarah mentions in his presentation. But also, as Sarah also mentioned, we can use also cell-specific questions, and we take advantage in the flow cytometry and the cell sorting, so you can select specific cells of populations of cells, and we can sort them and do single cell isolation. Then we can do lysis of these specific cells, and we can amplify their genomes using a repertoire of viral proteins. And so and we can obtain like mini genomes that sometimes can represent almost the 90% of a genome, so can be very useful, and is a very useful strategy when you don't have the possibility of isolate or even do metagenomics because the amount of starting material. But we have to let go back. So if we are talking about an oxygen gradient, how we can obtain that samples or study processes in an oxygen environment without having oxygen contamination, as Bo mentioned in one of the first lectures, or even more, how we can make sure that we are capturing the metabolic activity in the specific anoxic environment and not just transcriptional changes that are being generated from the sampling to the analysis. So normally when we are going to the sea, we deploy a rosette that have many different bottles, and the time that it takes from taking the samples to analyze them in surface is sometimes a couple of hours even. So if you want to capture the transcriptional activity, or moreover avoid oxygen contamination, you need to work with state-of-the-art technologies that allow this kind of analysis, and this was slightly mentioned by Bo. So we need to technological development for the study of this environment, and one of these equipments that we use and have used is the pump profiler system, or PPS. Everyone knows as PPS. That is an instrument that can pump waters directly from any depth that you desire, and it's also attaches to a CTD system, so you can go deep with the CTD, and you can select any particular depth as in example for us, the chlorophyll maximum depth, the secondary chlorophyll maximum depth, and you start the pump, and the pump start to pump water from that depth, and this is almost immediately, so you can have exactly the water from these depths, and you avoid a lot of oxygen constant contamination, and you can do transcriptomic without spending much time in recovering the samples. It's a fantastic instrument, and there are a couple of papers that shows very, very fantastic data in this paper from Macarena Troncoso and colleagues. They on board of Cabo de Hornos, I was there also. We deployed the PPS, the PPS who belongs to Osvaldo Ulloa's lab, and we take water from the pump in situ, and the group of Laura Farias, and they do a fantastic set of filters and membranes to obtain deep profiles and of nutrients and gases. This one of the first time that I saw some really nice gas profiles, and that each 1 meter. So I can show you here some slides from the paper, and you can observe the microstructure of the distribution of greenhouse gases. So these kind of tools are fantastic for working in really extreme environments. Also, I have the chance to work with the group of Beth Ward, and in this case with Luis Medina, who deployed also in the oxygen minimum zone an MS-SID, or the microbial sample submersible incubator device. That is a robot, or basically it is a mini laboratory, so you can ask any question, you can do incubations in situ, it can be able to fix samples in situ, and you can study whatever you want that live in the water column. So in this case, we use it to assess one of the first in situ assess of racing by protists in the OMSID. So it's a really huge instrument that use that voice that allows to put the MS-SID at the specific depth that you want to work. So it's also a magnificent instrument that we have the chance to use in one of the cruises that we go to the north. Of course, there is a lot of sampling strategies behind these works. For example, for single cell studies, that the one I will show you now, seawater has to be preserved in buffers that doesn't not alter the cell osmolarity, because the shape or structure of the cell is essential when you want to discriminate by flow cytometry. And in this case, this manuscript, we use single cell genomics for the study of indigenous populations of cyanobacteria that are the responsibles of this secondary chlorophyll maximum, and these cells have been recognized as oxygen producers in this anoxic environment. So these are really specific and indigenous cells that have many metabolic functions that other cyanobacteria doesn't have. So we demonstrate that there are basal lineage based on single cell genomics that have phenotypic properties of other cyanobacteria that are not an ancestral attribute. Additionally, we found that all these lineage that are indigenous encode genes for pigment biosynthesis under limited oxygen condition, a trait is shared with other freshwater and coastal cyanobacteria. And this suggests that Prochlorococcus, that the cells that we working on this paper, diverged from other cyanobacteria under low oxygen condition before the transitioning from phycobilisomes to transmembrane chlorophyll complexes, and probably they may have contributed to the oxidation of the ancient ocean. This is a really interesting paper that you can just download it for free from PNAS homepage. And this oxygen that is produced by this small cyanobacteria can follow other metabolism that can co-occur in this interface, anoxic aerobic interface. So there are many metabolisms that are really interesting to study together in these areas, and I will go deep in the presentation on that. This is another article that was just published in these days, and we use the multi-omics approach also, in which we combine single cell genomics with transcriptomics to study one of the key carbon degraders in the ocean that is recognized as one of the main carbon degraders in oxygenated environment, but at date, this is the first work that assessed their activity in low oxygen waters. And something very interesting that we discovered that they are really relevant in the secondary chlorophyll maximum, and these field campaigns demonstrate that the idea that Alteromonas can co-occur with Prochlorococcus and can help them in the carbon degradation and alleviate oxidative stress that many authors that demonstrate in vitro experiment occurs in the environment, and particularly it's occurring in this deep chlorophyll maximum. So also a very interesting discovery that we found using a combination of single cell genomics, and we observe here that there are some sacs here that are represented in the data set and combined with the metatranscriptomics when we were looking for a specific function of these cells. This is the paper if you want to take a look of in Frontiers. And this is something that we are working with many other researchers that in which has been a really hard task because we are having to develop our own tools for combining a lot of information. And one of the main findings, that is something that Sarah was discussing before, about how we can correlate the taxonomic profiles with the functional potential of the community. And I can imagine that any environment have differences in terms of the microbial structuring. That is a really relevant concept. And in the case of the OMCs or the oxygen gradient that is generated in areas where the OMCs occurs, the community is so structured depending on the oxygen gradient that we can capture this gradient in terms of taxonomy, but also we can capture it in terms of functional capacities of the community. This is a PCA that is based on merging three different data sets of barcoding data. When we merge here the 16S for bacteria, the 16S specific for archaea, and the 18S sequencing of small eukaryotes. This is all less than 3 microns, so we are talking about really of picoplankton. And we can observe a really structuring of the community based on the barcode data, and the 4 groups that corresponds to anoxic water, or the deep chlorophyll maximum in this case is in secondary chlorophyll maximum of AMCs, that is the yellow dots, that is the area that we are working on, and the chlorophyll maximum of the mixed layer, and the oxycline in gray. And this is a very ordered community or structuring. But when we're looking at the functional representation or the world community metagenomics based on KEGG orthologies, we can observe the same pattern of clustering and the structuring of the functions. That is really amazing that that the distribution is basically similar between the taxonomy and the functional capacity of the community. So this is a really powerful result that we have in the data, using a lot of metagenomes from the different oxygen gradients. And when we look for the models of concurrence, and we do a bar plot analysis with all the models, what mean a modulus of concurrence? It's mean that there are ASBs or OTUs, XOTUs, or we can do that like sequences that have similar distribution between the different environments. We can observe that secondary chlorophyll maximum or the chlorophyll maximum within AMCs have both the metabolisms observed in anoxic environment, but also metabolisms that are present in surface communities. So this concurrence modulus analysis demonstrate that the secondary chlorophyll maximum is an excellent site for studying microbial interaction and this interface between oxic and anoxic environments. So it's something that we are working on and in prep. And from this, we generate a lot of information. We have around 500 max that still have to be analyzed. So we are working on it with our own tools we developed with Salvador. Hope he's watching this presentation. A lot of pipelines to generate really nice figures and data. And finally, we can also not only work with taxonomy, no, we are talking about biogeochemistry on, so also we can work with single cell or single cells or community sorting to study processes in anoxic environment. So also with the PPS in the work that we were doing with Montserrat, and then she continued working on it. We take water from the deep chlorophyll maximum here in the secondary chlorophyll maximum that I show you here, where this Prochlorococcus and Synechococcus inhabits there. And we incubate the water with different tracers, and we measure a group specific based on single cell, based on sorting, nitrogen and carbon isotopic analysis. So based on this, we can also take an idea about those, and the results indicate that, for example, reduced forms of nitrogen are the dominant source for phycocyanobacteria. Alkyl nitrate may be important on some occasions. And also Montserrat in working with carbon demonstrate the delta 13 carbon from the POC is 3 per mil on average here, indicating that really is occurring photosynthetic activity in the top of the anoxic core. So we can even use these tools to study the biogeochemistry in terms of processes using fluorescence activating cell sorting. But also a part that I was not going inside in the presentation, but I think is something that is starting to be relevant is metabolomics, but particularly in this case, we use lipidomics, and we demonstrate that the IPLs or membrane polar lipids are really a good indicators or biomarkers or chemotaxonomic biomarkers for OMCs. And we can observe in this graph the depth distribution of IPL classes. So in two stations where that correspond to stations of Iquique that have this anoxic environment, and we can observe a depth difference distribution of the different lipids that belong to different microorganisms. And we can, and when we do a CCA for trying to demonstrate the structure of the community related to the environmental parameters, we demonstrate that the community in terms of lipid distribution also depends in the physical-chemical characteristics of the water column. So this reinforced the idea that also the metabolomics, in particular the lipids, are really good chemotaxonomic markers also for OMCs. So this is the paper if you want to take a look of the results and the use of lipids. So all this works together will allow us to integrate the ecological and biogeochemical knowledge in our work. So this graph just condense all the information that we are working on it. No, we are working with DNA, RNA, and not proteins, but lipids for phenotypes. And we can merge this information that is DNA based from whole community or single cells, and we can merge with physicochemical data that we are showing here in the workshop, but also with this experiment that I show you with carbon and nitrogen, and we can do this integration. And this is what we are looking forward. And this is one of the main objectives that we are working on CLAP with Alex and with other researchers. So where do we go from here? And the idea is to integrate the biogeochemistry with this sequence modeling oxygen minimum sum. So we want to determine how this interaction network forms, function, and change over time, and how we can expand this analysis that has been done by Loka and colleagues in an anoxic fjord. We will try to use the same approach to work in the OMCs from the Eastern South Pacific, that with data that we are being collecting from there. So you can take that data that we sample, we can model it with this kind of gen-centric models, and then you can establish the possible relationship between microorganisms and the functions that are present, and the metabolism and the reactions that are performed but that microorganism. So this is the one of the goals that we are working with CLAP. And there are last more ambitious objective, that is trying to crosstalk between these gen-centric models with bioevus. So this ambitious objective, we are working in collaboration with a lot of research groups in trying to incorporate the parameterization derived from these gen-centric models to implement it into the regional coupled physical biochemical model, that is bioevus, and sensitive experiments to boundary forcing and model parameters will be carried out to document coupled biogeochemical processes. That is the main objective that CLAP have, and Alex mentioned in the introduction of the workshop. So it is really a hard task, but we are a lot of researchers that we are working on trying to modeling this and trying to have a specific or site-specific processes that are occurring at sea. That's all, and thanks to all of you and all of those who have worked on the different articles that have been presented now, and Montserrat, Álvaro, Salvador, and Osvaldo mainly, and a need for the funding for the postdoc. That most of this work is part of the postdoc that I do with Osvaldo Ulloa at Universidad de Concepción and with the Millennium Institute of Oceanography, to the SEASA Center and the other centers that I have been working with. Thanks to all.
Thank you, Carlos. This is a really interesting approximation to the system. So I have a question, because this is really overwhelming, because these methodologies in molecular tools is growing so fast. So I remember when I started to study the biochemical aspects of the system, and the people was interested in track I don't know, genes to see if these I don't know, mechanisms are active represented in the genes, not just the taxonomic, you know, information. But now it's a lot of options, and the question is from from the people that is not to emerge in these methodologies. What is the best way to approximate? So is just to decide which you want to do? There is limitation not just for the processing, but for the taking the samples? You mentioned something about the time related to the RNA sampling, but is more than that, or is just easy to, for example, take water from the oxygen minimal zones and incubate it for doing some experiments to measure processes, and in parallel take samples from the RNA and processing in the way that you explained here, and which method you can choose? I mean, if I want to see if there is activity in about at the level of the genes.
Well, it depends. If you have a PPS like Osvaldo, you can do whatever you want. And if you have MS-SID, you can do in situ experiment, but no other groups. I think Ricardo Letelier and Osvaldo probably are the only group that have a PPS. So, and it's huge. You know, you cannot put it in a small boat. You have to go in a research vessel like the Cabo de Hornos. So you have to forget about transcriptomics. It's quite impossible to be realistic in having samples because it takes a time to deploy the rosette and take the water, and there's ways to create, and we are working with some technicians to trying to create a small pump that is not necessarily a PPS that have a CTD, and because you can deploy a CTD and select a specific depth and trying to have a pump that with enough power to pump water from the depth that you want. In that case, you can use metagenomics, but as Sarah mentions, that the single cell genomics is very powerful, and obviously you cannot cultivate almost any other, mostly any organism within an anoxic environment. So for single cell genomics, you need 1 ml of sample or 500 microliters. So if you have the technology or you have the equipment, you just have to put a weight in and deploy an iskin bottle and take samples for single cells, and in some occasions you can do metagenomics that also need 5 liters of water. So, and then with that information, if you can model the system, you can use this genetic information to try to reveal the processes and even the distribution of the processes. So that's the idea of the gen-centric model. And moreover, if you merge the results of the gen-centric with bioevus of these more general models for oceanic processes, will be fantastic, because you only need partial time samples, like seasonal samples, and you will have a really good description of the site, and you can start to do more specific question about rates as you're working on.
Okay, and about these single cells techniques, and how you what is the word that how you select, how you is more specific a word for that, how you select the organisms that you want to study, because I know that when the organism has pigments, it's easily, because
yeah, sure,
that, but for the heterotrophic community or the chemio-autolithotrophic community that has no pigments, which parameter you use, to select, to sorting is the word, to sorting the cells that you want to study?
No, for heterotrophic cells, most of the time people are just sorting a lot of cells. So this is a first round of amplification of the genome. So then you do a first round of PCR amplification of the 16S, and this gives you a first glimpse of which probably the probable taxonomic affiliation of the single cell. So then if you have like 400 cells and you have the taxonomy for the 400, you can, or an approximation to the taxonomy, you can select these cells. But by probability, the rare biosphere or the cells that are less than 1% of the community will be scarcely represented. So that is the bias of any technique have a bias. So probably with the single cell, you will have also the bias that you will select the most abundant cells. But in some cases, this is relevant because you want them. You want to select the most important cells, and you cannot have the ability to culture them. In example, we have I have like 500 max, and most of the max that we create with the metagenomes represent archaeas that are not culturable, no? And this is relevant and give you an idea of the potential of the community, but at the single cell level. So that's the main concern. But also now you can work with fluorescence proofs that, but you cannot fix the samples with something that froze the or froze the sample. That's why you use glycerol for preserving the cells for single cells, because you need to extract the DNA. So you cannot force the cells or inhibit the possibility for extracting the DNA and do the amplification of the genome. So there are many methodological concerns that you have to surpass for having high quality SACs, and Sarah shows some of this.
Okay, Carlos, thank you. Thank you for your presentation and for your time. So I'm going to present our last speaker. She is Dr. Candidate Dharma Reyes. Her research focus is on understanding the present and past changes in the hydrology of the East Pacific using stable isotopes, radiocarbon, and trace elements in benthic and planktonic foraminifera from marine and water column sediments. From marine and water column and in sediments. This approach reveals information about the water column and bottom water characteristics such as density, ventilation, relative nutrients content, and dissolved oxygen. Currently, she is in the last year of her PhD thesis in marine geoscience and working as a senior technician in biochemistry at the Lille Center, Heriot-Watt University in Edinburgh. She also is part of the Maroon Center in Marine Environmental Science of the University of Bremen. And she is also part of the Núcleo Milenio as well. So thanks, Dharma, for accepting our invitation and to be part of this workshop. And now you can start with your presentation.
Hi, everybody. Can you hear me? Yes.
Yes.
Okay, nice. Thank you, Alex, and everybody for inviting me to this nice seminar. I have been learning a lot, especially about microbiology. Today I will present something that is related a little bit, but not too much. I wanted to introduce you some of my results, my PhD that I named like mapping the ocean with stable isotopes of carbon, oxygen, and deuterium. Alex, if anybody have any questions when I talking, I think you could just let me know, especially related to methodologies. It's very important. I wanted to talk a little bit about me and my main goals in related to my focus of research. I'm very interested in trying to understand how was the ocean during the past, because it's the main key to understand the ocean today and how the ocean will behave during the future. Then, thanks to already 10 years of research since my undergraduate thesis, and I wanted to thanks to my mentors during all this process, I have been study sediments that could reveal us information about how was the ocean during the past. I have been also trying to elucidate how are the tracers that we used in the past and the behavior of them in the present day. Nowadays, in the research group that I'm working here in Edinburgh, we also wanted to combine all of these, and people that do modeling, climate modeling, can use the resources that we produce to understand how the ocean will become in the future. I think all my colleagues already present that one of the main focus today in this seminar is try to understand different sampling techniques and to try to check how the ocean behaves and different biogeochemical processes and how we study them. For that, I wanted to show you a very recent paper that published in Frontiers in 2020 that presents all the very nice and international collaboration related to how we could sampling the ocean, different techniques related to what we wanted to do. As Carlos said, that if we have a focus in, we wanted to sampling the microbiology community of the oxygen minimum zone. Now we have this very fantastic tool of pumping water with the same characteristic that we wanted to study the water there. And all these techniques have been not developing from one year to another, are decades of work and century of works. And thanks to the collaboration that we have in ocean science, we could develop all these techniques in different oceans and have information in these zones. Since this seminar is about techniques, I wanted to show you how we could take samples from the ocean. And I think already my colleagues present that, but I really like to show you how this works, especially for all the students that are watching this presentation at the moment. For a study any tracer in the ocean, oceanographers have been using this very basic technique that is sampling water with Niskin bottles. And this gives us the capacity to study, for example, the temperature, salinity, oxygen. Alex, Carlo, and other colleagues name about nitrogen, and also in terms of what are the focus of my research, I use the same techniques to study carbon isotopes, oxygen isotopes, and hydrogen isotopes in the ocean. And it's very important to mention that as a paleoceanographer, as a person that we studied the ocean in the past, we couldn't calibrate the process that we use if we don't know how the water column and how these tracers behave today. Then paleoceanography science is like, I'm in love with that because give us the possibility not to only study the past with sediments, also have a very good understanding about how the tracer works nowadays. And after to see this nice video that show us how people take waters in the ocean, I wanted to show you also like how we could understand a little bit some of the plots that I will show you today. They are super complex. When I asked to my family, hey guys, do you want, I wanted to show you my preliminary result of my PhD. And everybody was like, what is this? What is like colors in the ocean with dots? I don't understand nothing. Please explain this better to me. As you see here is a map that shows you latitudes in the y-axis and longitude in the x-axis. And in set, you could see the tracer that I'm in love with, carbon-13 of the DOC, dissolved in organic carbon. And in red or like more warm colors, you see enrichments of the signature of carbon-13. And in blue, you see depleted values. I don't want to really go in detail now in the beginning of the presentation of what is, but if we have more warm values, indicates that we have, for example, less nutrient, more oxygen in the water column. And if we have values that are like going to blue, we have more nutrients and less oxygen. And all the dots here that you could see is like, I will show this in a very didactic way. We're going with cruises to collect samples, and all the dots are different stations of where we collect samples. Usually we do in profiles. Here in this in the left side of the slide, you could see that, for example, are coming from 10 meters water depth. And then when we go, also we could like, what we do is like, we never have one cruise that could go through all the stations that I show you. Some of them, they go through, like this one here was going in all these latitudes, sampling water samples, but in Chile and in Peru, since the capacity in terms of financial programs and things, usually we could rent cruises in a specific time of the year and could go to specific areas, north of Chile, south of Chile 1 year, other year, central of Chile, other year, Patagonia. And then finally, after all the collaborative effort of these big scientific teams that collect samples, we could have like a kind of idea of how are the tracer in the water column. In this case, I only show you latitude by longitude, but I will show you in the next slide what is going on by depth. I wanted to show you like how we collect that. I already mentioned like already more than 2 times that we collect this using Niskin bottles. Carlos named rosettes. I don't know if someone in your home knows what is a rosette, but it's a simple, it's like we put all the Niskin bottles together, and in the way that the rosettes go into the deep, we could have all the Niskin bottle open, and when the Niskin bottle go up, we could close them in a specific water depth. This give us the idea of this, give us the opportunity in one like latitude longitude site to have by water column could be like 50 samples that reveals the entire water column tracer distribution. And also usually for have a rosette and this kind of equipment, we need a very big ship. Usually, for example, we could sampling that with the Cabo de Hornos vessel from the Chilean government, but also with international cruises that usually come to Chile and Peru once per year or every 4 years. But also, it's very important to mention that different working groups in Chile have been working with in very specific areas, for example, in Mejillones or in Coquimbo, and they use very small ships, and then the sampling techniques that they need to use is just like go with one Niskin bottle every certain depth and collect them one for one by depth, and require a very big effort, human effort, to take everything to surface and do all the sampling and stuff. Then it's very important that we consider that we need a very big collaborative effort to do this. And now we will go to see what are these isotope things. Like we have been, I think during the different talks, everybody has been mentioned isotope of nitrogen and stuff, but will be a lot of people at home doesn't know what are isotopes. To explain it very, very simple, isotopes are in the universe of elements that we have, and you know from the periodic table and elements in the nature, the elements behave depends of the number of protons and electrons that the atoms have. And what are isotopes are these elements that have different number of neutrons, and neutrons give to the atom the capacity of like have different behavior in terms of the mass that this neutron gives to the atoms. If I have, for example, an atom with one neutron and I have another atom with two neutrons, then these two elements that are the same with the same amount of protons and electrons will have kind different behave in terms of, for example, when the atom pass from a liquid solution to vapor solution, or very simple, for example, our cells, and when we like our cells perform some metabolic biogeochemical cycles inside of our bodies, and some of them prefer some elements that have less neutrons than the other ones, because for them are more easy to take them to do all the metabolic things. And then in this case, and we have some isotopes that have, imagine that you have a kind of element with an amount of protons, electrons, and then you have usually the elements to have a kind of equilibrium need to have a certain amount of neutron. If you have too much neutrons, usually the elements get very unstable, and these are the elements that we know as radioactive elements, and decay came to a more stable, and with neutrons that the protons and electrons are in the same kind of equilibrium radius. And almost all elements that we know from the periodic table are isotopes. And here are the very nice and colorful periodic table of the elements and isotopes of the UPAC. And the isotopes that we will talk today are hydrogen, oxygen, and carbon. Here is a very nice joke that I really love it. When we study isotopes, these are the isotopes of hydrogen. We have here like something that's very important to consider for the rest of the presentation, that you will see like kind of these numbers in the top left of each element are the mass number, and in the bottom we have the atomic number. And for example, here we have the different isotopes of hydrogen, and what we could see here that, for example, we have a hydrogen that have a mass number of 1, that means proton plus neutrons. Then we have deuterium that have 2 and 3. And what we could see here that, for example, here say, hey, I'm hydrogen. Then the people call me deuterium. Hola, mi nombre es tritium, y soy de Madrid. That means that, like what the joke is here, that these two ones, hydrogen and deuterium, are stable isotopes, and tritium is radioactive. Then usually all the biogeochemical part of this element and of this radioactive isotope of hydrogen is completely different than what we will see today. Usually, as I mentioned, and it's something that we really need to consider, that always the protons and electrons give the personality to the elements, and the neutron give a kind of how the elements feel during a certain period of time. It's like, for example, if I have more neutrons, I explained it before that I will be more heavy, or if I have less neutrons, I will be more light. And here is a table that explains different chemical properties of the hydrogen, deuterium, and tritium. For example, I wanted to take your attention in the melting point and boiling point of of these three elements. And what we could see here that, for example, it's not important which kind of isotopes we are. In this case, for example, we have protium, deuterium, and tritium, hydrogen 1, hydrogen 2, and hydrogen 3. And we could see that the melting point of the enriched element, in this case, the deuterium, is higher, and also the boiling point. And this like little difference between these two ones gives the possibility that we could trace different phenomena that we have in nature. Then, for example, here is a very didactic picture that explains, for example, what happened with in the carbon isotopes. It's the same with hydrogen and oxygen in terms of physicochemical characteristic. But for example, what happened if in a substrate with carbon 13, carbon 12, I go in a kind of energy hill, and what happened with the carbon 13 in comparison to carbon 12? Usually carbon 13, because it's a heavy isotope, usually need more energy and time to get up to the energy hill. And what happened with the carbon 12 carbon, is very easy that pass this energy hill. And in the product, usually I will have light carbon isotopes in this case. That means that it's not irrelevant if you are a light isotope of more heavy isotopes in terms of how you behave. In terms of the importance of the study of stable isotope as a tracer of water masses and biogeochemical process, Alex and the other guys already present that nitrogen isotopes are very important isotopes to study the biogeochemical cycle of nitrogen and carbon in the ocean. But like isotopes, and I put here the most important ones, have been very good studied in to study food webs or source tracking, rate processes, and the ones that I will introduce you today are the ones that we study in paleoecology and global carbon or global change. Something to try to explain you how we study the ocean in the past. For study the ocean in the past, we need to study sediments or different archives. In this case, in the ocean, we study marine sediments. And here's like a kind of example of how we take these sediments from the deep ocean. The first thing that we need to do is check the position or where you wanted to study the sediments. For example, if the main of the research that you are doing is study high-resolution sediments from the last century, then you need to go to the special location where you could find these sediments, for example, in the Mejillones Bay or in Calderas Bay in the north of Chile. But for example, if we want to study glacial interglacial cycles where we don't need very high sedimentation rates in the area, and we need just have few points that will indicate us one glacial cycle, one interglacial cycle, then I need to go to that portion of the ocean when the sedimentation rates are more low, as for example in the middle, like more offshore of the continent, and then I take the samples. Here we could see in a Sun expedition that we participated in 2010, how we could collect one of the sediment samples from the ocean using a device called gravity core. And here we are sampling with people from the laboratory different layers of that find in the sediments that give us the attention, because I don't know, we have like kind of different colors or different texture, and then for have preliminary results, we sampling them. Usually on shore, the things that we do is if we are lucky and we have time, we could sampling different portions of the sediment cores. But usually what we do is like we could describe the sediment core offshore, and then we take them back on land to do a very detailed study that cover from the grain size that we have in the sediment until stable isotopes or different other biomolecules, for example. In my area of expertise and the thing that we have been doing during the last years, we have been studying this little microorganism called foraminifera. We have some of them that live in the bottom of the sea, and some of them that are living in the water column. And in the end, the ones that live in the water column, where they die, they precipitate to the bottom of the ocean, and the ones that are living the bottom of the ocean, when they die, they sedimentate there. And these guys also, we could find them from in different environments, from like Atlantic Forum have been a very good study in the water column from almost all oceans around the globe, and benthic forums as well. Other thing that we could find in the sediments are molecules and compounds. For example, here are alkenones from these micro organisms called coccoliths. And at the same time, we could study the sediment itself, for example, taking some elements that are precipitated low oxygen as chromium. And also, this is not the only thing that we find in the sediments. As a microorganism, also we could find diatoms and other things. Then just to show you how the benthic foraminifera and the plantic foraminifera, I wanted to just show you like a very didactic thing about benthic forams. But people that study sediments, I will go back to this slide that explains a lot of things. People that study sediments, usually you could study geochemistry of these sediments and the geochemical part of the microorganisms that live in these sediments as foraminifera. But also people do things called paleoecology, where you could study, as Carlos and the other colleagues, the ecology of this organism. And nowadays, it's a very big effort in terms of trying to characterize the metagenomic of these organisms to try to understand their ecology. In terms of biogeochemical of this organism, we study the calcium carbonate. They are made by calcium carbonate, and they the main process of how they incorporate these things. The foraminifera are unicellular organisms that have a cytoplasm. And in these very tiny cells, they could do all the biogeochemical processes that we have been talking about today. And what they do is they take different components in the water column, for example, calcium carbonate that we could have it here. And they also incorporate other microelements, for example, magnesium or iodine or manganese, depends on the condition in the water column or in the bottom waters where they live. And they take all of them. Some of them they calcify their shell in equilibrium with the bottom waters and the water column. Some of them not. Some of them they have something that is called vital effects, that the organism have metabolic processes that take over the geochemical part that the foram will indicate us if it's in the equilibrium with the environment. And what we will talk about today is about, I will show you some of this disequilibrium in terms of foram with their environment, and we'll understand the different biogeochemical cycles that are operate about in this issue. Today, the idea is to mapping the ocean with these stable isotopes. And here I introduce you, like we already talked about hydrogen isotopes and oxygen isotopes. I wanted to introduce to you, and carbon as well. And in terms, something important when we talk about isotopes is their abundance in different components. And something that you could notice here, that the heavy isotopes like deuterium, oxygen 17, oxygen 18, carbon 13, the abundance of this element of this kind of isotopes are lower than the light ones. And this is very important because when we analyze these isotopes in the laboratory, this gives a very big, it's like puts to all the earth science a very big challenge in terms of how we could measure them in the lab in a very accurate way. I will start with talking a little bit about the oxygen isotopes. The oxygen isotopes and the deuterium isotopes mainly respond to the hydrological cycle. I think some of you already remember the hydrological cycle from your high school and primary school classes. Very simple thing, water in the ocean evaporates, we have clouds, and these clouds travel to the continental areas where they discharge rain, or if they go to very cold areas, they discharge snow. And the thing, when we study this hydrological cycle in the school, was very simple, but it's not so simple, it's very complex in terms of how the different isotopic composition of the different like reservoir of this hydrological cycle occur. And what we could see here, for example, the ratio between light isotope and heavy isotopes and standards. I will not go in very detail today about it. Called delta 18O in the ocean is around 0. And then what happened here, and as you imagine, is going to a very depleted values of delta 18O. This is explained because the light isotopes, as I explained you before in the previous slides, they are usually very easy to remove from the water through evaporation. And then what happened with the clouds? The clouds are gain more of these light isotopes. And in the end, the results is we have a depleted delta 18O value. And what is happening when we have these clouds going to the continent? I wanted to, that in your home, you could imagine the Pacific Ocean, Los Andes, and the coastal range mountains, because we will go through some preliminary results that we have from that area in the next slides. But then, for example, if we have these clouds traveling to the coastal range or the intermediate depression in Chile, we could see that the cloud is getting depleted. And then when, for example, if we go to more cold areas, getting more depleted. But what's happened here that the rain, if you could see here, if we compare the rain here in the first discharge and here in more cold region, the second discharge is getting also depleted. This is because when we have the cloud with the delta 18O values, when discharge like isotopes and also heavy isotopes, but in terms of the little proportion of these, usually you discharge heavy isotopes, and then the cloud that you could see is getting more depleted because I getting, like I doing the rain, all the rain is getting in proportion more heavy isotopes that are discharged and discharging. And finally, when I get into an area where it's cold or like high altitude, I have a cloud with quite depleted delta 18O and as well depleted delta 18O in the snow in this case. How we sampling the oxygen isotope of the deuterium isotopes is quite simple. It's super hyper simple. When we're sampling in the continent, we could sampling rivers, we could sampling the snow, we could sampling groundwater as well. And here I show you, and I will show you during the presentation as well. During my research, I have the opportunity to work with different communities of Peru, Chile, doing citizen science, collecting water samples for try to understand how the hydrological cycle works in this area. And here you could see one of our sampling campaigns in El Maule River, specifically in the Tenor region in the Los Andes. And we will collect the sample. You could see, we collect the sample, and then we need to, like, you could see that we have a range collecting the samples because it's necessary to filter the samples, especially for samples that are having some sediments or particulate material. Then we filter the sample and we save the sample in like a kind of cold conditions. We couldn't freeze the sample, or we lost the stable isotope composition, and the sample couldn't be evaporated because also we could lost the isotope composition. As I explained in the previous slide, the stable isotope of oxygen and deuterium response to hydrological cycle. Then the idea is we could keep the sample as a temperature quite stable, low, but couldn't be frozen and couldn't be evaporated. And we have been doing these campaigns in different rivers. Here are very beautiful photos from the Itata River that we went with my friends and colleagues from Conciencia Sur, from Los Andes in the portion of the Ñuble River to the Itata River, last portion close to the ocean. And like when Alex invited me to do this presentation, I try to be sure that everybody that are here in their homes attending the presentation will have access to different protocols that we have been creating during the last years, and are protocols that are open to all the communities that want to take these type of samples to characterize the hydrological cycle locally or more national, in a more national scale, then it's open to everybody. And Alex, if you have, or if you can, I think in the end of the presentation, we will give these resources to everybody at home to have the protocols. And as I mentioned, filtration is very important. This is not because, why if this oxygen and deuterium isotopes, like I just named it that need to be filtered, is because the machines that we're using in the lab usually are high sensitive to any particles that the samples have, and we could damage permanently some of the part of the machine that we use. This is the main reason. It's not because the particulate material that we have in the sample affect the stable isotope composition of deuterium and oxygen in the sample. This is very important to mention. And here we could see the machine that we use is a mass spec in the British Geological Survey here in the UK that we have been using for measured the different isotopes. And yeah, I invite you to also see the protocols, and if you have any question, we are always open to answer. And if also, like, since we are doing this citizen science like sampling campaign, also if in your community as a local student, for example, from different universities of Chile, you want to start to sampling like your groundwater or the water from your river or rainwater, just contact me and we will be in touch and you will be part of this very nice citizen science project. Here's some preliminary results that we have. This is the delta HNO values in the x-axis and magnesium aluminum in the y-axis. I will not go in this elemental composition of the samples, but just to explain you that also as a working group, we are trying to study different elements in sediments in the ocean and also in the continental part, and to try to understand different provenance of the sediments from the different rivers to the ocean. And this is the reason that I show you here this video. But I wanted to really get your attention in the delta HNO values that is going from -10 to 0 or 1. When we have very depleted values, as I mentioned in the previous slide, with all the nice drawings, we are close to like areas with high altitude or high latitudes. And usually we have something that is called the F members, that are the signature of stable isotope that's coming very close to the origin of the stable isotope composition, where in this case the water of the different rivers in Chile coming from. And we have a trend line. In Chile, we don't have rivers in Los Andes or in the coastal range mountains or in the intermediate depression that have values depleted of -30 or -40, for example, that are very typical from Antarctica or the Arctic continent. At the moment, what we have been founded in Chile, that we have values of -10 as the depleted value of delta HNO, and it's going to -5. Between this range, we have a different variability of values, but yeah, I think it's important to trust this kind of line of the maximum and minimum values that we could have of this tracer in the rivers of Chile. And what we could see here, that -10, with -10, we could find the signature of the end members of El Ñuble, Bio River, Calle Calle, Tolten, Imperial, Cautín, Maule, Itarta, Las Cruces. And then we have a range of rivers that for some of them, we thought that their main stable isotope composition close to the ocean is coming from Los Andes, but no. For example, we have Mataquito River, Mancu, Aconcagua, Tirúa, Lebu, Alonquén, that have values that we could think that are coming not from Los Andes as a main contribution of water in these rivers, will be like coming from the intermediate depression and the coastal range mixed with groundwater and also water coming from precipitate rain. These are very preliminary results. We are working in some areas where we have samples from Los Andes passing through the intermediate depression to the coastal range, but this gives us a kind of like light that we could use stable isotope composition of oxygen to trace the earth member of the different rivers that we have in Chile if we sampling the rivers in the last part of the river close connected to the ocean. In terms of wait, this, I think I repeated. Yeah. No, I didn't repeat. Sorry. To complement the results that we found in the rivers and in all the continents, we have been studying also stable isotope of oxygen and deuterium in the ocean. The protocol is the same that we use in the continental area. Sorry, my doggy. But in this case, we use our friend Niskin bottle, and we collect samples from different water depth, different position, and then we filter the water and then we analyze. And also, if you want to see the protocol, it's here, available to everybody. And some preliminary results here is from a paper that we published this year. Here in dots, we have the values of, we have the different cruises with different dot colors. And you could see that this is like a study of delta HNO and deuterium in the ocean range are very little. And this is the first effort of a very collaborative community from different institutes that help us to collect samples for these tracers. And what we could see here that we have an oceanic meridional section that here you could see here, and a coastal meridional section. The goal of this study was to establish earth members and also establish the typical values of delta HNO, deuterium, and carbon-13 of the different water masses present in the front of Peru, Chile. And what we could see here that in the oceanic meridional section, we don't have much results for these tracers. And it's very important that also give us the possibility and give us the reason that we really need to sampling this tracer in the oceanic section, especially because some paleoceanographic cruises occur in this area to collect very deep sediments, and like if we don't have any idea about the bottom water characteristic or the hydrology of the delta HNO and deuterium from the water column, it's very difficult then to have like a modern analog to compare with that. For the coastal meridional section, you could see here are almost like there are a lot of very regional sampling campaigns from different institutes of Chile and Peru. And we could see that, I don't want to go very in detail about the values, but red values indicate water masses more saline, and blue values indicate water masses that are more fresh. And what we could see here then in Patagonia, for example, and here in the x-axis we have latitude, and the y-axis we have water depth. All the water masses, I will not go in detail of them, but are named here. We could see that in Patagonia we have this fresh flow of water coming from Patagonia to the ocean, and then we have very saline waters that coming from the equator to until 30 degrees south in Chile. And then also we could see some subsurface water masses and some intermediate water masses that you could see that we don't have very good agreement of delta 18O at the moment. The sampling in this area had been very little. Then nowadays we are improving that, but with the information that we have, give us the possibility to statistically calculate the different earth members and also trace different water masses using delta 18O and deuterium. The same with deuterium, indicate the same as delta 18O. As I mentioned before in the different slides, the two isotopes give us information about the hydrological cycle. Then for oceanographers, for us, give us information about density, salinity, and temperature of the different water masses. I will go very, very fast in the next slide. Some other traces that we study is carbon-13 of the dissolved inorganic carbon. These isotopes give us the opportunity to trace the different reservoirs of carbon in Earth, atmospheric, surface sediments, intermediate surface ocean, intermediate ocean. We could study the carbonate sediments, organic rich sediments, soil, and different components of the carbon cycle. All components have different carbon-13 values. This gives us the opportunity to trace if, like as Alex also explained in his presentation, to explain how processes could go from surface ocean to intermediate ocean, how this could boil to sediments, and how these sediments, for example, have dissolution, and and what happened with the isotopes when these carbonates are coming back to the ocean, the water column, and so on. And I don't want to go very in detail with this because I think I'm almost at time. But to sampling carbon-13, the only agreement with the other samplings that I already showed you is the first part. We take the sample, the water sample, but then how we conserve the sample is completely different. The sample needs to be poisoned by mercury chloride that give us the possibility that all the life that is in that sample is like death afterwards, and the sample need to be taken without bubbles. That means that we don't have contamination of the atmosphere in the water sample. And in the process in the lab is very rigorous and strict. We need to first put all little vials in vacuum, and then fill them with helium. Afterwards, when we have this, and before that, like I think you could see here a little bit, it's not very clear, but we put phosphoric acid, and then we do all the vacuum and put helium. And then afterwards, we inject the sample. And what happened, we have all the inorganic carbon going to gas as CO2, and then we analyze the CO2 gas in the mass spec. Thanks to this method, we don't contaminate or dissolve inorganic carbon sample with any organics that coming from the process. All the process occur in terms of preparation in low temperatures, and the acids start to dissolve all the calcium carbonate present in the sample, and that will be dissolved and particulate. Also, you have the protocol there if you want to see it. They're available to everybody. And here we have more results. Also coming from the same publication that I mentioned before. Remember, all the dots are the cruces, and what we see here in this slide, we see in the left side the carbon isotope composition of dissolving organic carbon in the ocean setting here, and in the right side the coastal section. And as I mentioned before, I didn't went very into the cycle, I think it's because of time, but if we have high values of carbon-13 indicate as like low nutrients, high oxygen, and when we go to low values of carbon-13 of the dissolving organic carbon indicate as high nutrient, low oxygen. It's not always one-to-one, but in general it's like that. And what we will see here that all the colors that you see are the different water masses. Some water masses are characterized by low carbon-13 values, or for example, the equatorial subsurface water and the subtropical waters in the north of Chile, as well Pacific deep water that we could find it in the deep ocean in almost the entire Pacific. And these water masses, as I mentioned, are, like also Alex mentioned, are water masses with low oxygen and high nutrients content. And you could see extreme values of carbon-13 here. That is like this dark blue is areas where you have anoxis processes occur in the water column as metaionogenesis and other processes that you could have very depleted values of carbon-13 in the water column. And all these red colors that you could see here indicate water masses with high oxygen, low nutrients, are the typical, the ones that coming from Patagonia and from the subantarctic zones here, and are the Antarctic surface water and the Antarctic intermediate waters. And the big things, or the main thing that we found in this publication, that usually in paleoceanography reconstructions, we have been using for more than 2 decades the results that coming from the oceanic section that you could see here, that with my pointing, I'm covering, because was the first expeditions that gave us a very nice characterization of the water column in the Southeast Pacific. But what we found that the water column structure in front of Chile and Peru, close by to the coast, are different, and almost all the sediment cores that we have been collecting by different cruises for the last 3 decades are coming from the continental margin. That means that we have been using a modern analog that are not I don't want to say a wrong modern analog, because was the information that we have during that time, but a modern analog that could be give us another kind of vision about the water mass structure. And this is a very important point, because I went through this mapping, or like the title of the presentation was Mapping the Ocean. As I mentioned, to study different sinking sediments. For example, here we have sediment cores in black dots that covering, or that give us the opportunity to study the different water masses in the past in a very high resolution in terms of space. We have different samples that covering a specific water mass and also the mixing zone between the water masses that you could see here in dashed lines. Then what happened if we don't have a very good understanding of this modern, as I mentioned here, this modern distribution of the water masses nowadays with the specific carbon-13 values of delta 18O or deuterium, is quite impossible to really give interpretation of how the ocean was in the past if we don't have this very clear in the present days. And especially if we study these little organisms that I mentioned, foraminifera, that are made by calcium carbonate and other trace elements, if we study the isotopic composition of oxygen and carbon of these forams, we could find, like, and these are some preliminary results that are, like, we wanted to send it to publish in the next couple of weeks. Here we have a carbon isotope composition of different species of these foraminifera that live in the bottom waters and in the surface sediments in the entire Southeast Pacific. And what we could see here that is here in this, in the right part of the slide, we see the difference between the foraminifera and the water. As I mentioned before, usually when we want to have a tracer that are similar to the water, we select foraminifera. Their calcium carbonate carbon isotopic composition is similar to the water. And this gives us the indication that we could use them as an indicator of carbon-13 in the past. And what we see here is the anomaly of this foram versus water. And we could see that some foraminifera have depleted values, like if when we compare with water, and some of them are quite in equilibrium with the water masses where they calcify. And one of them, that is this one, that is Cibicidodes willestorfi in red dots. These are the, like, I will say, the goal X of paleoceanography. Everybody around the globe uses this species because we know that it's calcifying in equilibrium with the environment. But in front of Chile, specifically in the oxygen minimum zone, horizon between Antarctic intermediate waters, we have a disequilibrium of the value of this species. And that means that is something that we need to consider, especially for paleoceanography reconstruction, and try to understand if these values is coming because the foraminifera have a kind of different metabolism or adaptation to the oxygen minimum zone that we know that have low oxygen, also is quite acid, low pH, and then will be the forams have this kind of adaptation and calcify their values in like almost 1 per mil more than the water. Then to show you why it's important, these are sediment cores in front of north of Chile, and what we are doing is like a kind of end close of the presentation. We have been compared surface sediments with long core reconstructions that give us, for example, the capacity to study the ocean like very past, like thousands of years ago, and like here you could, like, some preliminary result of, we compare delta 18O of the forams, carbon-13 of the forams by age, and that we could see that we have, like, we have been analyzed more than 60 gravity cores from different locations that give us the possibility to really reconstruct the waters. How was the waters during the past in different water depths and latitudes in the full continental margin of Chile, Peru. And here you could see, like, I wanted to just show you the colors. Holocene, that means foraminifera that have been living during the last 4,000 years, and last glacial maximum, a period of time 23,000 years ago, where the ice with the ice in the mountains part and in the continental part of Antarctica and in the Arctic gain the maximum volume at the moment in the Earth, very cold periods. And what we could see here is, for example, carbon-13 values of this foraminifera indicates, if we compare Holocene with last glacial maximum, that the water mass are like here above 2,000 meters water that are quite similar. And also in the north part, like in the equatorial Pacific, are quite similar to the Holocene. But what happened here in the south Pacific, close to Patagonia, we see that we have depleted values of carbon-13 in this sediment cores that could be indicate that could be a water mass that are quite depleted in carbon-13. That means could be more anoxic or like more depleted in oxygen, and will be have more nutrients arrived in the south of Chile during this period, but was very resilient and have very strict latitudinal range until 30 degrees south and didn't go far to north during this period of time. And these are the things that we are studying with our working group right now, and these are the results of my PhD thesis. And what we are trying to do now is we are like all the things that we are published now are open to everybody. We are using Python to create all the plots, and everything is available in our GitHub of the laboratory. If you want to go through that, just write me an email or just ask and I will send you the things. And all this work like have been like a collaborative effort of a lot of people that you could see here in the photos. Some of the people is here participated in this seminar, and as you see, if we understand how the ocean works today, is very like a very important question to understand how the ocean was in the past. And if we understand how the ocean was in the past and how it's today, we could give this information to our nice friends that doing modeling to understand how the ocean will become in the future with the different CO2 scenarios that the IPCC present us nowadays. Thank you.
Okay, thank you, Dharma, for the presentation. Definitely, isotope signatures are a really interesting tool to approximate to the study of the variability of the system. Now we have space for the closing words of our research director of the university. Sí, sí, por favor, vamos con el video de las palabras de cierre de la directora de investigación, por favor.
Research on oceanic processes has not been sufficiently visible in the study of the climate change impacts at a global level. This is translated into a lack of tools, technologies, and protocols to understand the role that, in particular, the minimum oxygen zones play, especially about microbial communities and how they are being affected by the climate crisis. On behalf of the Universidad Católica del Maule, I would like to express my deepest gratitude to the researchers who have generously shared their work and who, due to their responsibility and compromise, have decided to create a Chilean-Danish biogeochemical network to understand the impact of climate change in a coastal upwelling ecosystems. This network will provide the scientific community more and better evidence that contribute to propose effective adaptation and mitigation mechanisms against the consequences of this new climate reality. I wish you all the greatest success in this collaboration that is being consolidated in the challenges that you will have to face. You will always be welcomed and supported by our university. A big hug to each one.
Thank you, Dr. Zúñiga, and thanks you all for attending this event, and thanks to my colleagues for your time and for these interesting presentations. I hope the topics discussed have been clarifying the investigation that each of you in the public developed. I also want to thank the communication team of the Catholic University of Maule to support and develop this activity. And finally, we hope to come back with you soon to show you more results from EnviroNet. Thank you, everyone, and have a good rest of the day.